table and a quick show of rules.
Turn yourself down a little bit.
It's on channel 1, like that.
I'm
Good.
Juliet, I've been told to come at this time.
Hi Juliet,
I just get a couple of things out.
what's your name?
Mikey.
And please introduce yourself to microphone.
Hi, I'm Juliet. I have been told to come to start.
Anyway, so this is Talkaoke. It's a pop-up talk show where we talk about whatever you want to talk about. Have you got anything to kick off with? Any thought?
Obviously, the question is, why Star Wars? Everybody keeps asking me.
Why Star Wars? Yeah.
Why Star Wars here? That's what people keep asking me, and I reckon it's because it's Brian and it's in the stars. That's what I've been
So
telling people.
you think it might be related to Brian Cox? Why Star Wars? Have you got any views on the Star Wars thing?
Or is it because they're gonna sequence the DNA of Chewbacca?
Okay,
I want to find out. We
Yeah.
tried to get Chewbacca on the table yesterday, unsuccessfully.
We were having a mammals conversation, and I just put it to Chewbacca.
That's what
Are you a mammal? Are you interested in mammalian? Are you a mammal? Come and join us. This is a whole mammals conversation. So I tried to bring, just like that, I tried to bring, in fact, exactly like that. And there was a connection because I think Chewbacca was kind of saying, yes, I'm a mammal. But we never got any further than that.
The only other thing I could think of is that on the International Space Station they do biological work.
Yeah. Okay.
You know, they do a lot of.
I mean,
bits
rather than an X-wing fighter, should we have like a replica of the International Space Station? Do you think that would be more of a draw, or more relevant maybe to genomics?
No, it's fun. It's good.
It's
Science
fun.
is fun. It's important to keep science fun.
Keep science fun. Are you a scientist?
I am, yeah. Well, yes, I am. I am a scientist.
Was
I've
it the
got a
fun?
PhD.
Was it the fun element that got you into science?
Yeah, and the mad people.
Yeah. Okay. So it was. Do you find science fun?
No.
No? Why not? Come and join us. We've got Margot, we've got Ellie and Mikey. I mean, just tell us. You don't find science fun. I just want to.
I actually am missing it all.
Okay. All right. But come back. We'll be talking about something else later on. Do you find science fun?
I don't know enough about science, but I know. I know a
I'm
scientist.
going to put that down as a.
But no. But as you were talking about the space, what do you call it? Space base up in.
The International Space Station.
Yeah, exactly. My friend, whom I met for doing comparing gigs at Polish weddings.
Yeah.
He's the DJ. He is also working. He's a biologist, so he always ensures that whatever goes up to the space is completely clean, or there's got everything that they want to test and stuff. So he's got quite a responsible job. But you wouldn't know it from knowing him as a DJ, as I did. So I worked with him for years.
He's a DJ and biologist.
Yeah, so I think that this is his way of trying to keep his life fun. That's what I think, because his work is very serious.
Okay.
Biologists are the most fun.
Biologists are the most.
I must go back because I'm meant to go
Okay,
on
give us a final.
working.
What are you doing?
So I'm at Miltenny Biotech. I've been there 22 years. That's how fun it is there.
Yeah.
So yeah, no, it is. It's good. It is good fun. You meet really lovely people. I work with good people. You work on things that are important, but equally so you enjoy doing it, and that's what matters.
Was it called
Miltenny Biotech.
Miltenny? What does it do?
We do anything to do with cells, so it's like separating out cells from different tissues, through to then checking what cells you've got, so using flow cytometry, growing them. And it goes within 5 years of the techniques being developed. Max separated cells are in patients, and we have a clinical arm. We do all sorts. We've got imaging, all sorts, anything to do with cells and biological research.
Okay, I'm going to hit you with a round of applause. Oh no, that's the wrong one. It's this one.
All the best.
Thank you, Ellie. Good luck. Maybe we should have have I got a clinical arm? Both my arms, and neither of them are clinical. Yeah.
I mean, it's good to not. There's a saying.
Yeah.
No, it's a hand, not arm.
Yeah.
One arm doesn't know what the other one is doing.
Yeah.
So maybe you do.
Yeah.
But you just don't know it.
Okay.
It's the unknown known. Unknown known.
I would say if either arm is more clinical, it would be the right arm. Yeah. Anyway, that was Ellie. I personally think that having a next year, this is one for people watching this, it's not been like this all the time. There's been more people before, but I think having the International Space Station replica, I've seen that. There's one at the Space Centre in Toulouse when we did a gig in Toulouse. They've got one. So maybe have a replica of the International Space Station next year for a Festival of Genomics, because there is actually a genomic link. Yeah.
Maybe I could get my DJ friend to DJ a set. Like last night I was on social media and I saw, is it Jesus the Redeemer in Brazil? The big statue of Jesus.
I don't know what it's called, but I know what you're talking about.
I think
in
it's
Rio.
Jesus exactly.
Yeah.
So Jesus the Redeemer, they had a priest there doing a DJ set, and it was really great. And this is a festival. I'd like to see a DJ set around the space agency.
Okay, should there be a DJ set here? I mean,
I would say so.
we've heard from Ellie that biologists are fun. Maybe they would like to have that maybe they would like, or even like some DJ, like a DJ lessons, how to mix tracks together. It might be an interesting thing for next year's festival.
I mean, would you then have to kind of liken the DJing process into sequencing, or like we press this button, this comes out, or
in a way, it is a kind of sequencing, isn't it? Come and join us. Yes. Come and join us. This is Talkaoke. We've got Margot. I'm Mikey. We're talking about what would make next year's Festival of Genomics more fun. Would you go to a DJ set, for example? No? What would make it more fun for you? Yeah?
Hello. But I think it's the question of music. Like, when you run a festival, I mean, I wouldn't go to, for example, as an example, I wouldn't necessarily go to a reggae festival, but I'd go to a techno festival. But then, is there specific type of music?
You wouldn't go to a Reading festival, you'd go to a
reggae. Reggae music.
Reggae festival, yeah.
So there has to be some kind of a link that brings people together, and I wonder whether there's a common type of music for people who are scientists. Is it genomics beat? I don't know.
There might be I've got a feeling that like physicists might be more into techno, whereas biologists might be more of a kind of house, go for a house vibe. But we need to speak to some people to confirm or deny this. So we just need this person. Hello. No, I think they're in too much of a rush, but maybe this person. Hello. I've got a question for you. What would make next year's Festival of Genomics more fun?
I've only just got here, so I'm not sure, but I don't know, like more coffee places or something.
More
like
coffee?
That. Yeah. Just give me a week.
Maybe we could get into the coffee more, like actually it could be like a coffee brewing workshop or something like that. Would that work? Because I'm obviously, biologists are into process quite a lot, so they've probably got a lot to say about how to brew a good coffee.
No, but is it obvious how coffee impacts different people? Like, if you drink a coffee and I drink the same type of coffee, the same amount of coffee, is it obvious that we're going to be impacting the same way? I'm just curious because I don't know.
Is there a genomic element to coffee sensitivity? Maybe. Thank you for that. More coffee. I think I've had enough coffee, to be honest. But anyway, but is there a genomic element to coffee sensitivity? Come on. Yeah, 2 minutes. Yeah, well, don't worry. I've got eyes on you. If anyone comes, I've got eyes.
Just for information, yes, there is a genomic element to caffeine sensitivity.
Okay.
So some
Come
people
and sit down
digest.
and tell us. Yes.
I will when my colleague comes.
All right. Okay.
But yeah, there is. So you've got genes which essentially digest caffeine. Some people, it will wire them to heck because they don't have it and they can't digest it, and it'll go to their bloodstream quick. Some people have a great tolerance because obviously their genes are built that way. So yes, you can actually be tested and go in there and actually test to see how it will affect you.
Go in where? Is this done in Starbucks? Where are they doing this research?
They'll do it in Boots the chemist. They'll do it in Lloyd's the pharmacy.
Really?
You can buy caffeine sensitivity tests, just like alcohol dehydrogenase, just alcohol, the same thing. Anyway, so no, less than 50 quid.
Okay. But that's a lot of cups of coffee, 50 quid. Thank you for that, Paul. So we're talking about what would make next year's Festival of Genomics more fun. That's what we're talking about at the moment. And we've got Margot over here. Tell us your name.
Matt.
I knew that, Matt, actually. But what do you think? We've come up with a few different ideas for next year's festival. One is, any ideas for next year's festival of genomics? What would make it fun?
I like the TIE Fighter, the X-Wing, Star Wars X-Wing. So maybe, I think that's good, that's fun at the event. Maybe get some other kind of characters that people like from films.
Okay.
And also Switch, you know, where you play computer games, virtual reality headsets, Oculus Rift. Yeah.
Okay, that's a lot of good ideas. Hello. Any ideas what would make Festival of Genomics more fun next year?
If it's going to be Star Wars related.
It doesn't have to be Star Wars.
It could be anything.
It could be anything.
Oh, right. No, you put me on the spot, really. I don't know. I'm sorry.
I'll tell you what we've got so far. Oops, I don't know what happened there. Some other ideas. So far, we've got a DJ set, a coffee brewing workshop,
yeah, at the work.
or the International Space Station.
Yeah.
Yeah, that'd be very good.
Cocktail making.
Cocktail making.
As the after evening networking.
Yeah. Okay, so make your own cocktails.
Yeah.
Can you trust biologists to make their own cocktails?
Yeah, because then you'd get really good, inventive, creative cocktails, some different ideas.
Okay, like genetic cocktails.
Genetic DNA cocktails.
Yeah, okay. All right, thanks for that. Thank you. Thank you, person on the phone. We're talking about what would make the Festival of Genomics more fun. We've got Matt, we've got Margot. Tell us your name again.
Inez.
Inez, that's right. I'm Mikey. And so far, we've just had a great idea. Cocktail, instead of the drinks being just beers, could we get biologists to make their own cocktails? Would they be good at it, or would they be spectacularly bad at it? And what about not just cocktail mixing, DJ mixing as well? These are some of the ideas. Inez, have you got any thoughts on that?
So I actually hosted one of those like make your own cocktails for the group nights with some of my friends, and we did it based on our jobs.
Yeah.
One of us was a pharmacologist, I'm a biologist, so I do DNA extraction, so I called mine the Genomic DNA extraction.
Yeah.
So it was gin,
Yeah.
and then I added like what was it? A
Could
raspberry,
we have Festival of Genomics?
Yeah, exactly.
within Festival?
Genomics. Like
Yeah.
the pun's right there.
Yeah.
It was red, so it looked like it was blood, and then I used sweets to make it look like a little thing of DNA to garnish over the top.
Okay.
And it gets people engaged, and it's fun, and I think also it's the kind of thing people would take photos of it, share it online. Come to the Festival of Genomics.
Okay. Sorry. Matt, any thoughts on this? What would make? Would you go to a genomics workshop? And what would make the Festival of Genomics more fun?
I think if we combine it with the tests for alcohol sensitivity, I think that would be pretty fun. But I'm a scientist, so I'm very biased. So for me, knowing things is the most fun thing of all.
Yeah. Knowledge is fun.
Knowledge is fun?
Yeah.
Is knowledge fun?
I don't know.
It's boring,
is it fun
right?
for you? I mean, what's the most fun bit of knowledge for you?
The most fun bit of knowledge is how proteins fold, because I'm a protein scientist.
I mean, could you sort of like make a macroscopic sized protein out of cardboard and
that's what people did. So, you know, and back in the 70s, you know, when the structures were discovered for the first time, where like X-ray crystallography was a thing for proteins. You had like these massive structures built to explain the structure of hemoglobin, for example, or myoglobin, which are proteins that carry oxygen in your blood. That's hemoglobin, and myoglobin, that's the protein that stores oxygen in your muscle.
Okay, and so you had giant cardboard structures that people could fold up. Maybe that would be, we're talking about what would be a fun activity for the next year's Festival of Genomics. And would it be genomics? That's what Inez is suggesting. Could it be like, could it be like a cocktail mixing workshop instead of just the normal drinks. Matt, being a protein guy, I know that's controversial in itself, but could you have some kind of origami workshop with large protein structures that you could fold in real life? What would make it a bit more hands-on, a bit more fun? So we've got Margot, we've got Inez, we've got Matt. Tell us your name.
Hi, I'm Abby.
And
Kevin.
Kevin, welcome. So yeah, any thoughts on what would make next year's festival more fun?
I got an idea. You can use a DNA extraction kit, like a peel from an onion.
Yeah.
And just see it under a microscope or something like that. And it's easier to do. No mess
so
about.
sort of like DNA live, so you're actually hands-on, get people, and maybe you could even show it to some of the bioinformatics people or people that have never actually seen organic material before. Yeah, okay.
So we just need a microscope and onion peel, that's it.
Okay, microscope, onion, it sounds appealing to me. We've got some new faces, so we've got Matt, we've got Kevin, we have got Abby, and we've got Inez, and we've got Margot. Tell us your names.
Emilia.
And
Iksan.
What was it?
Iksan.
Iksan, yeah. So at the moment, I'm Mikey, by the way. At the moment, we're talking about what would make next year's Festival of Genomics, or would it be Genomics, more fun. We've got some really, really good ideas, from cocktail making workshops to protein folding to just getting a microscope out and getting some onion peel under the microscope. That sounds appealing to me. So that's what we're talking about at the moment. Kevin, have you got anything? Any thoughts? I'll let you think about it. Anything on... Oh, Matt, yeah.
I think extracting DNA on its own, probably, because this is a genomics festival, it's full of geneticists who just do it every single day.
Yeah.
That sounds like bread and butter. I think it would have to be like a workshop. It's directed at patients, you know. You could maybe make them take their own tests for genetic disorders.
Their own genome, rather than send it to...
Or make their own gene.
Yeah, make their own genome.
Exactly.
Yeah, okay.
Synthetic genome.
Okay.
Craig Venter.
We can make our own organism for next year, like a festival special. That might be an interesting use of yeah.
I was just wondering, because there's a lot of scientists, not so many patients, even though arguably everybody's a patient as well. Yeah, but there's not many. But I wonder whether, how you've got BYOB festivals, bring your own bottle. Can you do BYOP, bring your own patients? So everybody brings a patient with them. So you book 2 tickets essentially for yourself, and you have to bring a patient. If you don't, you can't come. And then that way you're mixing up, the conversations, you're mixing up stuff. So
well, let's find out. Let's just do a little bit of a litmus test here about whether people would be able to bring a patient or a research subject or somebody. Would you be able to bring anyone along?
I mean, I don't have patients at the minute. I'm still a student, but I'm very interested in sort of the translational bit from research to patients. So I believe, because it's just getting more and more into our daily lives, I think having an aspect that's more about communicating that science is
so
really
how do you actually translate that research to real people? Yes.
Because I work in a biobank, so I work with kidney disease patients.
Yeah.
So
could you bring a patient along?
I could actually.
You could actually. You could. Okay. Would they be willing to come to this event, especially if we had free cocktails?
They're working with us for the research. They're giving blood samples and data for us, so they'd be happy to help us out.
Okay. I mean, maybe giving alcoholic drinks to people with kidney disease may not be the best thing, but maybe the protein folding they'd enjoy. Kevin, have you got any thoughts?
I can see the idea of bringing the patients. I mean, where I work, most of the time in the lab, so I'm not really patient-facing, and what I deal with is mainly patients with single gene disorders.
Right.
So some of them, trying to get them in might be of a challenge, because we do tend to go through a lot of rounds of GDPR and things like that, so you have to...
I mean, is that a surmountable obstacle? Could you get over that? Could you somehow, through all these kind of like GDPR or other kind of data barriers, bring people in?
I can see it, but it obviously has to be full consent for the patient. So I mean, that's the other barrier you have to get through.
Okay. I mean, would they want to come? Do you think in the end, would they be curious?
I think some patients will be curious, especially the ones that really want to know what's going on with their health and the innovation that is working towards getting them where they need to be in terms of therapy. So, yeah, I see why not.
Yeah, okay, so there might be some interest. Okay, so where do we want to go with this? Ines, did you have something to say? I'm quite interested in this, the translation question. Maybe we could delve a little bit more into that, but Ines, what were you going to say?
Well, so we actually, I'm at Newcastle University. I work mostly in the labs as well. But we host an event. I don't think it's hosted by us. I think it's like a collaboration thing with the NHS and everything. I can't remember what it's called exactly. It's like Genomics for Everyone or something like that. But it's patient focused. So we have speakers that are coming who have rare diseases, and they talk about their experience and what they understand from the stuff that we do and the stuff that we research. But we also host little workshops. So I do one of those like extracting DNA from a strawberry, and like the public can get involved with that. For a lot of people, it's strawberry? It's very easy, but you just mash it up in a bag and use like.
Is it because it goes well in a daiquiri?
Yeah. Well, yes. I used to be a teacher, like a school, secondary school teacher. So I did it with my year 11 class or something. But it's all household items. So it's strawberries and then like dish soap and salt, and then like alcohol to. Get it all, and it's the first time for a lot of people actually visually seeing DNA. So I think that I guess that's part of the translation, that it's not just DNA isn't just this like weird concept. It's a real thing. We're like it affects all of us. And the questions that we end up getting from the public are so different to the kinds of questions that you would get from an academic setting.
Can you give us an example?
Well, they want to know actually like how long does it take for these things to develop, like what's the instead of asking things like what genes of interest, what's the significance of these genes, they're actually asking, they're like, well, what does that mean for us? What kind of treatment is that going to end up being? And they want to know actually what is that for someone who has a rare disease, rather than us thinking, oh yes, we can then like market this and further research this and make a paper. Like that's kind of the end goal for a lot of academic research stuff.
Okay, so it's a totally different focus. We've got a new face on the table. We have got, I'm going to start on this side, Margot, Ines, Abby, we've got Kevin, we've got Matt, we have got Amelia and Iksal, that's right. And I'm Mikey. What's your name?
Maria.
So we were talking about what would actually make the Festival of Genomics more fun, and we come up with lots of different ideas about actually how to bring in like a wider audience, a wider perspective, a patient perspective, but just general punters that coming off the street. How do they understand DNA? You can mash up a strawberry, mix it with some dish soap, bit of salt and alcohol, bang, you've got DNA. You can actually see it. And stuff like that, but we're coming up with some very fun ideas. That's where we are right now. Matt?
So I was just thinking now that one of the coolest things would be making a dinosaur, like, from the Jurassic Park, but that's impossible, right? Let's be honest, we cannot make a dinosaur. But what we can do is we can genetically modify organisms. So how about we do like a 2-day workshop?
A genetic modification workshop?
Yeah, we make some glowing stuff. There is this protein GFP that just fluoresces, so it's beautifully green and looks sick. First day, people just take a piece of DNA, put it into bacteria. The next day, bacteria grow, and bam, they're glowing in darkness. And that's pretty cool, because, extracting DNA, sure, we all got DNA. It looks a little bit gooey when you extract it, blah. But
actually, you want to go further with it and
let's go further. Let's make modified things, because that invites people to think about how programmable we are.
Okay. All right. What's that?
Bring your pet to work day and make it glow.
Bring your pet to work and make it fluorescent. We had a whole pet discussion yesterday. But
cats are better than
our dogs better than cats, or cats better than dogs. But what about what is easier to make fluoresce, a dog or a cat?
Could we, as part of this workshop, could we just create some kind of like a cat dog, and then please everyone? Could that be a workshop?
Would it be a dat or a cog?
Well, exactly, I don't know. But could we like mash it up a bit, make it glow on top of it?
Okay, so do some real-life genetic experiments. Could you combine a cat and a dog, and what would it be called? And would it be fluorescent as well? These are some of the questions. A lot of questions to ask, but you know, we want to hit people with a lot. Is these the kind of questions that your people at the Genomics Day in Newcastle are asking?
Not really, but maybe that's what we should be asking, because it's kind of limitless here. So you know, I'm sure there's a thing later that's like trying to pitch the best like AI function of something, and like you never know what kind of ideas people have until you have something like this, where I think when people are made to be open to sharing their ideas and not afraid that it's going to be shut down. Like it's even if it's just for fun, you know, like we're not feasibly maybe going to bring a cat and a dog and fuse them together, but the ideas are there, and it's what can be done, what is actually possible within, you know, our field of genetics here.
I mean, we came into this discussion because we were asking people, why did you get into science? Is science fun? And most people, not everyone, said that. Yeah, science is fun. I love doing science. But, you know, in a way, this discussion is about how do we actually, okay, we can isolate a gene, but can we isolate the fun and actually show that to people, show the real passion of being into these things? We've got a new face at the table. What's your name?
My name's Jacob, sorry.
Hopefully you heard that little intro of what we're talking about, what would make next year's Festival of Genomics more fun. How do you actually isolate the fun that scientists have when they're doing experiments, when they're doing their research? How do you actually isolate and contain that and show it to people that are maybe not scientists? That's what we're talking about at the moment. We've got Inez, we've got Abby, we have got Maria, we've got Kevin, we've got Matt, we have got Amelia, we've got Ixal and Margot and Mikey. Yeah.
You know, so in terms of showcasing that, maybe try and integrate AI into it and come up with these simulations. It will be actually, I think it will be a lot easier in terms of rather than having someone hands-on doing it.
So you can actually do this with AI. Exactly. You don't actually need to create a cat, not a cat, a cog or a DAX.
I mean, that's where you start before you can go practical, really. I mean,
okay, so could we be using AI? I'm going to come to you in a second, but these two are going to go, so I want to get a final thought, and that's it for everybody. If you do leave, just give us a final thought, because I'm sure Amelia's going to have a great final thought.
Yeah, I have a thought about AI, actually, and I really had it last night. I was having dinner, I thought, scientific innovation, you know, when I was born, things already existed, so I just took it for granted. I was like, someone was there, someone tried it, it worked, great, we have it now. I'm in this position now with AI, and I'm honestly kind of scared about it. I'm like, oh my God, what could this mean for us? But I've decided to, that's very cynical.
That's scary.
But I've decided to embrace it. So, you know, when my kids are born, maybe AI will already be implemented in the clinic, and they will trust it because they're like, someone has tried it out. So I'm embracing AI, and with that, I'm going to go on a talk and listen to some more stuff.
To be honest, this year, people seem to be embracing. Last year, everyone was feeling guilty about using AI. This year, everyone's like, bring it on. We don't know what we're doing. It doesn't matter. AI knows. So that's where we are. Ixal, have you got a final thought for us?
Oh, yeah. I think the most thing that we can do, I'm a gynecologist that working in genetics as well, is to attract the mothers, because mothers will bring a lot of people to the conference. So if you want everyone to know about the genetics, bring all the mothers.
Okay, so mothers are the ones to target. How would you target the mothers?
Yeah.
What be your advice to bring in those mothers for next year?
Well, what do you know about your kids? Like when you get pregnant, do you know about your genes? Yeah. You can attract a lot of mothers in there, and they will talk to their community.
Okay, so in a way, it's like talk about your children. Tell us what's special about your children would be the thing to bring in mothers. I feel like I've kind of ignored you on this side, but thanks very much. Any thoughts on any response to any of these things? Should we use like real life DNA to bring people in, to get people excited? Or should we go for the AI visualization? What do you think would be more effective?
I really liked what you were saying about like bringing the experience, not just of the excitement of science, but of the process of doing science at the festival as well, and kind of getting to see a little bit. I think people often feel, which was maybe hinted at there, a bit of a disconnect from like, okay, what is? There's lots of people here who do lots of different things. What does a day in your life actually look like? And I think that's one thing that can be quite mystifying. Like, what is the process of doing science actually looking like? And I guess we get close to that with things like kind of posters and research talks, but it can still feel like, I would love to see like a bit about what people's days look like across the like vast variety of kind of roles and like topics that we have.
Okay, well, you've come to the right place at the right time, because we can just do that now. Let's do it. I mean, I have to say, as an artist, people, you know, we've got a saying that art is 1% perspiration and 99% administration. And the day in the life of an artist is mostly writing emails and proposals. But I'm sure for scientists it's more interesting. But what is and don't go for like a really dull day where you're just, you know, you're just trying to correct the references on your paper. We don't want that day. We want a more fun day. What is a fun day for you, whoever you are as a scientist and artist? I'm going to start off, you propose this idea. What's a fun day for you?
A lot of it. So I am a data scientist at a startup. So we've got some people who work in the lab, and we've got some of us who don't work in the lab, and we know very little about what, we know relatively little about what goes on in the lab. So we're kind of like sitting often in what you would kind of imagine a very data sciencey type thing. We've got like some sort of spreadsheet open or kind of some coding terminal open, and we're kind of like just making like pretty pictures out of this until there's a point at which we go, we haven't got a clue what's going on there. Why is that blue when it should be red? And then the most fun bit is then when we get to walk across to a building, across the street to the lab, and go like, we don't have a clue what that's all about. And then every single time we do it, they tell us something that seems incredibly obvious to them that we didn't have the slightest sense.
You're like, explain this, and they actually do explain it.
Yes.
Okay.
That's what
all right. Okay, we've got a new face over here, but we're going to go round on the table on this one. So we've got Margot, we've got Jake, we've got Inez, we've got Abby, we've got Maria, we've got Kevin, we've got Matt, we've got
Molly.
Molly, welcome, and I'm Mikey. I'm not going to hit you straight with what is the most fun day for you, but we're going to go around the table, so we're going to come to you probably last, apart from Margot. Are you ready to go now?
Yeah.
Okay, go.
I thought about it while I was listening.
Yeah.
So I'm a lab scientist, so I'm the other side of the table, if you will.
And a fun day for you is when all the data scientists come in and go, what's going on? And you can tell them.
Yeah, it's interesting. I don't work like directly with data scientists. I do a lot of my own data analysis. So I'll like have my own spreadsheets. I'll like organize. I love like slotting in what different tasks I have to do in the lab like in a day, because sometimes my experiments run over like several days because I work with bacteria, so they have to culture. And then I do like presentations, like go to a seminar, give a talk, that kind of stuff. I have meetings with people, and yeah, when I'm in the lab, I'm just like doing pipetting. I'm in like a cabinet doing like, you know, culturing, inoculating stuff, or working with some machinery to like measure something specific about what the bacteria are up to.
What's the most fun element of that? Is it the pipetting? Is it the growing the cultures?
Yeah. I think it's probably the organizational side. I really like the slotting and like also like presenting the data to people and like making really cool slides and making it like a really visual like representation of what's been happening in the lab to make it like understandable by everyone. I think that's probably my favorite part.
Okay, so it's like the results day. I'm going to come around, Matt. Tell us what a fun day is.
A fun day. So it's a bit because I'm an extrovert and I like talking.
Yeah.
It's explaining things to people.
Yeah. So you must have been having fun today.
Oh, totally.
Telling us about P53 again and again.
And again and again. Cause that's the best thing about teaching at university, because you go into a classroom and, you know, for a full hour, people are locked with you in the room. They have to be there. They have to listen to you.
So a captive audience, basically.
Yeah, essentially. So no, but my point is that I think something that I do try to practice is the storytelling of science. I think that's what's missing. I teach a lot of people, and I think the problem is that we as scientists come off as like really boring people. You know, because we just go on about things that are like sometimes really in depth. You know, talk about some kind of numbers. Like, who cares? You know, like.
So it's more about the story. You need like some drama, some actual.
You need some drama. So like, I'm just gonna test out the story on you because I actually need to do. I actually need to do that. So listen to me.
Okay, make it a quick one.
It's gonna be a quick one. It's about the science I've been doing. So. imagine that you're playing Waldo, finding Waldo. Do you know this game? It's pretty hard to find Waldo, right?
Yeah.
And imagine now trying to find Waldo, but you don't know what Waldo looks like.
Okay.
That's pretty tough. And that's what I've been doing as a PhD student. I've been trying to make a P53 protein, a Waldo, for other scientists to be able to spot it with other tests. Did that story land?
I think so. I think that was great.
Awesome. Okay, I'm gonna carry on using it. Thank you.
I think in the UK we call it "Where's Wally," not "Where's Waldo." But anyway, apart from that, I think you definitely get full marks for that. Okay, Kevin, fun day.
Fun day. So I work mostly in the lab. I'd say I'm more of an introvert.
Yeah.
And my life, I mean, you get to play around with DNA a little bit.
Yeah.
But it has to be purposeful because these are patients. So most of the time we work with lab scientists to try and get results out. So on a typical day, I will come in, see the patient, I will see what they have and things like that, and then you'll be like, okay, let's see how we can try and help you today.
Yeah.
It's kind of like, because that's why I go into science, to try and help people. So I look at it from, it's fun because I've always grew up doing like silly experiments, playing with firecrackers. That's how it all started, but then I transitioned into more of the biology side.
So is it actually for now for you the helping people, the fact that people, is it the understanding of people when people come in with their conditions, or is it more about the practical help? What?
I think it's a combination of both and trying to find meaning in that. So I mean, I would look at the results as well, bit of analysis, and you'd see, oh, they probably have this mutation, which translates into a clinical condition, and then you're trying to do that correctly, and then we work with the scientists, and obviously they push that to the patient themselves. So, yeah. That's my typical day, I can say.
That's great. I'm going to come to you in a minute, Matt, but we are doing a go round the table thing. So I need to find out, there's 3 people we've got to do still, so...
Yeah, I won't be that long because I'm a voice therapist, so I'm not a scientist. I'm not a researcher, and I don't really know why I'm here. No, I'm here because it's very, very interesting to me. I spend most of my day working with voices. Voice problems or no voice at all, and starting to think about voice as biomarkers. So for different conditions, there's quite a bit out there about mental health, and also other types of conditions that you can have with your voice box.
I mean, when it comes to AI, because yesterday we heard that AI is now recognizing babies from their voice, what they're actually trying to say.
So interesting.
Could AI actually recognize the kind of pathology of a voice by just listening to it?
Yeah, so some of our colleagues have been looking into that, definitely, and they're definitely seeing promising outcomes from that. So that's kind of what I'm interested in, so that's why I'm here. But yeah, I'm really enjoying listening to everyone's backgrounds.
But just as a voice therapist, is that what you are, a voice therapist?
Yes, I work in the NHS.
What is a kind of good day for you, or a fun day?
A good day, I think, is a breakthrough day, getting someone's voice back.
Right, yeah.
Really.
That must be amazing.
And it's really incremental.
Yeah.
It's a very slow process because there can be all kinds of things factoring in. Wow. Lots of acoustics and getting up and doing lots of physical things. So that's a really good fun day for me. Thank you.
Some voices. What's that?
She has a great voice.
You've got a great voice. Okay. What's a fun day for you?
So,
Abby?
so I work with gene therapies as well. So we experiment with frog, the tadpoles.
Right. Yeah.
So explaining it to other people.
Yeah.
So, and they're calling us cruel because we're experimenting on animals. It's cruelty, but we have to say it's better than doing on humans because you can euthanize them before they have a consciousness. So we can study what they're made of, like what are the effects of this gene, and then just end their misery.
So what are you doing to these frogs and tadpoles then? And how is it fun? Or can you not say it's fun because you're actually experimenting on animals, and you're not really allowed to say it's fun?
It's not fun, but
yeah,
it's not moral, I feel.
Yeah.
There's still a tadpole.
Yeah.
It's a different topic of debate. How
Well,
moral
it is. We can go there with Talkaoke. We can go anywhere we want, really.
But we have to think of humans too. How we can help the human generations.
Yeah.
So explaining it to other people. It may sound cruel, but it's needed. We have to do it.
Okay.
To study.
Can you say a little bit about what you're actually studying?
So one of the gene that's responsible for human growth, or how do I put this? I'm not really allowed to talk about this, but
just change the names to protect the innocents. It was not a frog; it's a toad, for example. Yeah.
So sometimes the human brain doesn't develop properly.
Right.
So what gene's responsible for that? So it's similar to a tadpole.
Yeah.
So
it's about developmental genes and how they kind of sequence. So
if you delete that gene, the frog might not develop properly, or without eyes, or just deformed.
Yeah.
So it's really cruel.
Yeah.
But then we can know what gene's responsible for that, and then pass it on to the researchers on higher health who's working on humans.
Okay, I'm going to hit you with a round of applause. We might, if there's some questions, maybe we can do questions later, but we've got to incorporate everyone's questions into what, we can go, it's up to you, we can go, I know you've got a question, Matt, but we can go down any avenue collectively we want to go down. But Inez, I want to find out what's a fun day for you.
I'm going to take little bits from everyone there, because I'm also quite extroverted.
Yeah.
So I love a good yap. Something like this, I think, is really fun.
So this is a fun day.
The chance to be like, guys, this is what I do, and these are the things I know. Knowledge as well. Like, I think for the most part, people go into science because they have this like thirst for knowledge. They really love finding things out that other people don't know.
Yeah.
And we're all big kids at the end of the day. Like, you know when you would get a chocolate bar and it would have like a fun animal fact, like stuff like that I think is really interesting. I didn't know this stuff about frogs, but I would love to know more about frogs, because then I can go and like tell my friends, like, I found out this really cool information about tadpoles, and like just really, I think if everyone has that, like, I know it's really cliche, but like, what's the most interesting thing that you do? But like sharing that around, I think we just love to be able to like share information with people that you think is really interesting. And breakthroughs as well. Like for me, I think a fun part of science is when you get that thing that you've been searching for, and you get your end goal that you've been wanting that whole time. There's a real high to that. I guess in the same way that like an athlete would get a high from winning a competition. Like I would find this gene I've been searching for, and that's like the best day of my life, because that's you've been working so hard to do that. But even the small wins day to day, like, oh, I've extracted like this many patients, and then new things, like I do long read genome sequencing, and it's like once I get past a certain number of terabytes of data, I'm like, oh, I've never had this much data before. This one's really good. Just really small wins like that. I think it really like, but it's really nerdy. But I think loads of people are like this, and I think we will have that conversation with people. It's really nice.
Yeah. How many terabytes are you on right now?
Oh, like we've got about 16 terabytes in one patient, which is insane.
Wow. Okay, so I'm going to bring this over to Matt, because I know you've been wanting to ask a question for a long time. It's probably become completely irrelevant by now, but
no, it's not irrelevant.
Okay, good.
No, it just struck me that as scientists, we tend to talk about problems from the science perspective quite a lot. So we're just like, we start stories by saying like, oh, there is this gene, there is this protein, and I'm very guilty of this, because I'm just like, there is this protein P53, but I realize, like, I like talking about science overall, and I like listening to science. So, for example, how was vitamin B12 found out? Do you know? I'll tell you. So, like, about 100 years ago, there was people going into hospital and they were like dying of anemia, but their iron levels were like quite good in the blood. It just didn't make sense. They were just dying. And what they found was that if you give like lightly browned liver to these people, 1 liver a day they would have to eat, they would be absolutely fine. No one knew why, so people started extracting things from liver, and they isolated these these complexes, the intrinsic factor with the vitamin B12, and suddenly it turned out that you can just inject people with that little molecule and they're fine. And they do have a genetic disorder. They lack the intrinsic factor, which is produced in stomachs,
right,
which binds vitamin B12 and transports it to the liver. But otherwise, we wouldn't know, because if patients weren't dying 100 years ago, you wouldn't know what vitamin B12 is.
Okay, thanks for that. We've got a new face to the table. We've got Molly, we've got Matt, we've got Kevin, we've got Maria, we've got Abby, we have got Inez and Jake and Margot. Tell us your name.
Eva.
Eva and Mikey. You knew that. We're talking about, we're sort of like, first of all, we're talking about what's going to make next year's Festival of Genomics more fun. And then so we're talking about that with some great ideas. Festival of Genomics, cocktail making workshop, science in action, extracting DNA from strawberries and making strawberry daiquiris at the same time. So we're talking about all these, and then somebody hit upon the idea that science itself is fun, and people get the reason why people actually got into this in the first place is once upon a time they were actually having fun. So we asked people what actually they enjoy about their day-to-day work, and you've missed that now, but yeah.
You're not sort of doing like Star Wars ideas. I mean,
we started off with Star Wars.
Like maybe Star Trek next year.
Star Trek, I've got a button for that.
You've done that.
But then we moved on to something that what is more biologically relevant, maybe the International Space Station, or maybe actually scientists in action doing real genomics here live in the Excel Centre. Molly.
Strawberry daiquiris.
Nice. Yeah. Okay, so we've got some Festival of Genomics people are watching this, not now, but later on. Give us a final thought, Jake.
One thing, this is from a while back, but when we were doing kind of round the table things that we enjoy, things that make a really good day, I was very kind of moved and interested by the people who were talking about kind of like having done amazing things like interacting with patients, having kind of like been able to see someone make progress in their kind of journey towards restoring or reclaiming some of their voice. Actually, one that overlapped with me a lot was just like, I can't remember your name, sorry, but you mentioned just like making a really nice picture, like just some like, I think there's something about like, I don't know, it will come as no surprise from me visually that I'm the kind of guy who loves a graph, but like just making something that like this both visually looks great to me and also somehow like feels like it has by its construction solved the problem, like a question that I had. I think there's nothing quite like a really good picture that's aesthetic, but also kind of somehow meaningful, and then you can go and like show it to someone else and be like, look at that. So that, I don't know, that I enjoyed hearing about that too.
Okay, well that's good, and maybe we could get people to bring their graphs next year. Bring your graphs next year. We'll probably have forgotten the whole discussion, but it still will be interesting. Abby, are you off as well? Give us a final thought.
My final thoughts would be get more scientists.
More scientists?
Yeah, get them younger. So make it like a get your kids to work day. So get them interested, get them engaged. That would be more helpful.
I mean, do you think we could actually bring in like school level scientists or undergraduate?
Interesting story. That's how I started.
Right.
I saw a live DNA extraction when I was in 9th grade.
Right.
So
was it a strawberry?
No, it's actually onion peel.
Okay, the onion peel. Yes.
Yeah.
We forgot about that. Yes, you mentioned the onion peel.
Yeah.
And
so
that's
how
actually
I got
what got you
interested
into
in science. And becoming a scientist. So whenever any of my relatives ask, what do you want to become in the future? I'm like, scientist. So I'm here. Made it.
And that's the story from now to there.
But no, just thinking about scientists, non-scientists, speed dating, speed networking, in mentoring, inspiring, ideas sharing. I really would appreciate something like that.
I mean, you said you're not actually a scientist.
No.
You're curious.
I'm definitely curious.
A voice therapist.
Yeah.
What kind of things are you curious about? What kind of things would you like to know when you wander around this, or are there unanswered questions for you?
Well, I think it's just demystifying a lot of it because it definitely seems quite mysterious from the outside, as a clinician, just sort of seeing patients day to day, thinking about data and what I can do with that because I've got loads of patients.
Yeah.
So it's just thinking about how to make things better for them, and putting my mind to work or putting AI to work as well.
Yes.
So I'd love to know more.
Okay, so maybe it's about how to look at that from a different perspective, from a genomic perspective or a data perspective.
Sorry, I'm away now, but just on the note of graphs, I think there should be a best graph competition.
Yeah.
Like most aesthetically pleasing, but also maybe like the best actually like ones that people can understand the best or something. So not like the same categories, like they're all graphs, but
you know, some different categories.
Yeah, you can have different ones, like the most confusing graph even. But also like images, like best image competition.
Yeah.
And it could be a whole range of things. It could be kind of like staining and stuff down the microscope, but it also could be kind of maybe sometimes a data set just looks really pretty, or like genes being mapped can look quite nice, especially when they're all color coded. So just like a best image thing, and it just gets people engaged. It's not necessarily something they need to understand. It's just something that they want to get involved with, and I think that then helps to engage people. It might trigger them to maybe ask a question about something that they don't know about previously.
Yeah, I mean, over here we've got like a lot of stuff which is from, I think mostly from patients, younger patients. But maybe we could also have like a gallery of graphs, a gallery of images next year. Thank you, Inez, for that. We're here all day, so come back later on. Thank you, Maria. One question to...
Sorry, I just really thought, is the King's Speech, the film, is that real? Is that how you train people? You annoy them and you make them talk? Or
Sometimes. For the most part, no. But that is one of the fun parts of the job, definitely.
Yeah.
We could do that.
Okay, so it's sort of slightly related, but slightly...
Yeah, a lot of acoustics. I mean, people lose their voice for varying reasons. Maybe they talk a lot. Or maybe they don't talk enough. That is also an issue. So, yeah, it's kind of lots of things possible. I do hypnosis. I do all kinds of things. It's quite fun, yeah.
Okay, so each case is different. Oh, each case is different, and each case has a different treatment in a way.
Yeah.
And the King's
Speech.
The King's Speech. I have actually seen the film, but
his arms around and things like that. We absolutely do that, especially with the kids. So I work with kids and adults.
Okay.
Yeah.
Okay, so there is a legitimate treatment. It is based somehow on the science.
But it is not elocution lessons. That's what we get. That's what we get a lot. It's not that.
Okay, so it's a bit different from elocution lessons. We've got a new face at the table. We've got Kevin, we've got Matt, we've got Molly, we've got Eva, we've got Margot. I'm Mikey. Tell us your name.
Hi, I'm Finn.
Finn.
At
the moment, we're kind of talking about, well, I feel like we're kind of transitioning a little bit. We were talking about what makes science fun, how do we... Okay, we've captured the data, we've captured some of the new insights from genomics, but are we capturing the passion that scientists have? And that's what we've been talking about. And we've heard about what makes different scientists tick and Maria over there is a voice therapist, so it's not just scientists. Yes, Molly.
I just had a thought thinking about the graphs thing, because something that someone else mentioned, like a graph competition. I feel like I think about art a lot because my partner's an artist.
Right.
And I feel like with art, it's about connecting to human minds, or like multiple human minds. It's about portraying like an element of what's inside your mind to someone else's. So that's what art is in my mind. But with scientists, I feel like we lack that connection.
Right.
So it's like we could take a lot more from the artistic community to try and make those graphs more absorbable by other human minds. So like, I don't know if we could invite or include graphic designers or like 3D artists or like, and I do not want AI art to be included in this conversation.
All right. Okay, that's...
But I just think it would be really cool if scientists could like incorporate more aesthetics.
Or could there be a kind of workshop, artist-scientist workshop, where artists actually collaborate live in the festival? Any artists or graphic designers around the table? Anyone?
I mean,
Yeah.
Well, I'm not one, but maybe there is another person who is. No, but my background is performance, so I wonder whether there's something that you can.
Yeah, because we were talking about how much people got from standing up and saying their results. Maybe we can actually do a performance workshop of like
or like poetry or something.
Yeah, how to really deliver the results in a performative way.
Yeah. I agree with that. Thank you.
Okay, so more artist collaboration, be they graphic artists or performers. Finn, any thoughts on any of this? I know you've only just come here, so you probably got no idea what we're talking about.
Well, if we're talking about making graphs and things more presentable and lovely, things like that, I quite like the fact that if anyone does single cell RNA sequencing at all, the package, one of the premier packages is called Shirat, after the artists who made plots or made paintings that look a lot like the UMAP plots that they make.
What's the name? What's the name of the
Shirat, is it George Shirat, the artist? Shirat. My art knowledge is nowhere near as good as it should be, but it's interesting that there's definitely obviously that knowledge of art that people would like to do, but a lot of plots are very dull.
Yeah.
A friend of mine loved making monochrome plots that were very, very difficult to read in lab meetings and things. So I definitely think some sort of training on like how to make visually engaging plots.
Would you like to know a bit more about visual engagement? Or yeah. Okay. That's interesting. That's it. Okay, so Chirac. Anyone here heard of Chirac, the artist Chirac?
Do you mean Seurat? Yeah,
Seurat.
Georges Seurat, as in the pointillist.
Oh, Seurat.
Yeah.
Okay. Okay, you've heard of Seurat. Okay. Any thoughts on this?
Well, I come from an arts background, and I work for a museum that has a Seurat in the collection.
Okay, you've got a Seurat in your collection?
Not in my personal collection, no. But in terms of like collaborations, artists, scientists, I think it's all about audience and communication. So, you know, why are you doing it? Are you doing it in order to make things look nicer? Yes, you know, but then who is it for? And I think one of the issues today, sitting in a lot of the presentations, is that the graphs were not readable from the back. Now, I know that sounds like an odd thing to say, like, but, you know, if you have a really, and that is a case of not knowing.
So you've
not
got
what
some
your
issues
audience.
with the,
Well, no, the problem is not knowing who your audience is, who you're communicating with, and or how big the screen is, you know, things like that, and actually how, but I mean, even in smaller settings, you get people, you know, with tiny, unreadable, too much text, etc. And I'm not going to just rant about PowerPoint or anything like that, but I think the point behind it is to do with knowing your audience. Like if there was there are some art science collaborations, but again, it's who is the beneficiary? Is it, you know, is it ultimately to create an artwork to engage the art community about science? Is it about creating an artwork to engage the science community about art? And you know, obviously there's some space in between, but it's all about identifying who's this for and what you want to gain from it.
Okay, I mean, asking the audience, maybe you could just ask the audience if they can see it at the back. Can the people at the back see this? Anyway.
I have a thought. I think that's a cool idea. I think we don't need like artists and scientists to collaborate and make a canvas or a poem. But I agree with you on the PowerPoint stuff. Like, we need to understand visual aesthetics. We need to understand readability, like color choices, the text size. Like, I just don't think that we think enough about aesthetics, not just for the like pleasant element of it, but in terms of what is the graph trying to communicate.
But you wouldn't want to make a poem, though.
No, but like, I just mean it's not just like, let's make art. It's like, no, how can we take like important visual elements from art, from visual art into science communication and science talks?
Any thoughts on this? I'm going to come back to you in a minute, Eva. I just want to, Kevin, have you got any thoughts on this? Or Matt, have you ever felt like you'd need the services of an artist to actually improve the kind of,
I think what would be cool is if we could engage all 5 senses when communicating science. I think we're so used to the visual and audio aspect of communicating science. So like, there is the graph, there is someone talking about it in a monotone voice, but have you ever tasted the graph? Have you ever smelled it? Do you want to smell it?
Yeah. Okay. I mean,
would that work? I'm just asking. It's just out of curiosity.
You're
if people...
a protein expert. What does your protein smell like? Has it got a particular smell?
I don't know. It's it smells a little bit because of the buffer that it's in. It smells like old eggs, but it's not the smell of the protein. It's the smell of the buffer.
Okay.
I don't think that the protein smells, but it would be nice if it did.
Okay.
I think maybe I can just go on another, maybe to add to his point, or maybe go in a slightly different direction. I mean, because when you look at art, you're looking at it 2D, right? Okay, so maybe incorporate 3D, or maybe make it more audiovisual, for instance.
Yeah.
So you can maybe try and animate that to make it make more sense. Maybe, for instance, I mean, because we're human beings, you know, we have to be visual. You have to have the audio as well.
Yeah.
You have to have like the speech as well in that element. So I think maybe in that sense, if you try and animate that and create some sort of user-friendly way, or because I myself, I'm not that creative enough.
Yeah. But I think it's an interesting point you made about connecting with a human being.
Yeah.
How do we actually make it designed for a human being to appreciate?
And because most people interpret things differently. Some people are more visual, some people are more audio. Like, for instance, I'm more of a speech type of person, so that's how I can process information better rather than looking at it.
Okay, interesting. So. Okay, so I'm going to I'll come to Eva because you're desperate to say that. I'll come to you, Margot.
Something that Molly was saying made me think also about accessibility, and actually, I mean, you talk about all of the senses, but we also have to reach out to people and actually legally generally have to, like, it's not like it's not just the right thing to do, but actually legal kind of requirement that we make certain things accessible to everyone, and that might and therefore there should be kind of minimum requirements when it comes to certain things like PowerPoint presentations, quite frankly, because regardless of the fact that you're at the back, even if you're at the front for some people, if it's below a certain font size, etc. So just bringing in accessibility and the importance of that.
Okay, so as Kevin mentioned, different people can access things in different ways, and there's preferences, but there's also just abilities to access information. But also there is an artistic side to this as well, and an appreciative side to this as well. Come and join us. Okay, after the meeting. Finn, any thoughts? Oh yeah, sorry.
Oh yeah, I just wanted to say that maybe, you know, maybe there should be like an art curator or something. Let me just very quickly say, maybe there should be like a PowerPoint artist curator or something. So it's not only are the PowerPoint standardized, but they adhere to all these maybe different requirements, like accessibility, and they are creative and nice. And I also wonder whether, I hate virtual reality goggles, but maybe that's something that can work for people to engage with the graph. You know, there could be that.
You could sort of travel along the graph or something.
Yeah, or something like that. And then the final thing I'm going to say is that in terms of engaging different arts, maybe there could be, I mean, I don't quite know how proteins fold, but could there be like a giant slide or something for people to jump on to have
earlier
fun?
on, I'm not sure if you were here, Matt was talking about doing a kind of origami protein folding session, which you could actually feel the shape of the protein. What's that? I'll find the thought from Kevin,
Also, just to add, I know I'm leaving, but maybe onto that we can also incorporate like virtual, like a VR headset, for instance, and maybe project that.
Would you put your head into a headset, and what would you like to explore with virtual reality?
I see why not. Like, I'm not exactly saying the virtual reality aspect. I'm saying maybe if you have data or information and make it more artistic, for instance. You can do that individually for each person. And
okay, well, thank you very much, Kevin, for dropping by. Where are we going to take this conversation? Do we need to stimulate the creativity, the presentation of some of this data, make it more interesting? You rejected the idea of a poem. Do other people think poetry has a place in science? What about painting? What about music? What about all the other arts? Matt?
I think recently someone told me about this AI that just makes TikTok videos out of like textbooks.
Right.
And like a lot of like medical students, like it was a medical student who told me about this, that apparently you just upload a textbook and there is some Minecraft video and there is like robotic voice that tells you about, I don't know, diabetes or whatever from medical field, and it works really well. So maybe, instead of reinventing the wheel and making a giant slide for a protein, which costs a lot of money, how about we just make TikToks,
Okay, mate.
It's a TikTok presentation, just stop presenting, just upload the TikTok, everyone can just watch it in real life, and if everyone likes the more likes you get...
Are you on TikTok, Matt?
Of course.
Have you ever put any of your work on TikTok?
Not yet, no. I always wanted to, but I won't. I would have to like sit down and think about it, and I don't have the luxury of always doing that.
Yeah, well, maybe we can help you. Are you on TikTok? That's what we're talking about now. Is that a good place for science communication? Are you on TikTok? Come and tell us about it. What would make a good... Matt wants to bring his science into TikTok. But it requires too much brain power. That's the problem. So we need extra brains to help him. He's a protein specialist. The protein is P53. But how does he actually bring this into TikTok? That's what we're talking about now. And have you ever seen any science on TikTok? We've got Matt, we've got Molly, we've got Eva, we've got Margot, we've got Finn. I'm Mikey. What's your name?
Florence.
And
Daisy.
So you were on TikTok. Do you ever see any science on TikTok?
I do. I actually follow Scientific American on TikTok.
Yeah.
So I see like news stories or like scientific takes on current affairs and things like that.
And is the TikTok a sort of particularly different environment to, say, other ways of communicating that science? Do they do it in a different way on TikTok?
Yeah, totally. I think it's much less formal. It feels much more personal. And I think it's also quite interactive, so it's quite like lots of people will comment on it, so it feels like a bit more of a dialogue, and you can hear other people's takes as well. So yeah, it's much more personal, I'd say.
Okay, that's really interesting. Are you on TikTok as well?
Yes.
Anything else you want to add to that about how science is?
No, it just needs to be short and snappy, and you just need to have loads of content like already prepared before.
Okay, I mean, I think that's the issue that Matt's at. That's how do we actually chop down these complex ideas into something that's TikTokable?
I think there should be a requirement. Like, if you publish a paper, you should publish a TikTok.
Okay, so
about the paper. Like, if you want to get funding, make a TikTok about it.
No TikTok, no funding. Okay, yeah.
Oh,
are you on TikTok then?
I had TikTok for a bit, but I had a problem with the app where if you hold your thumb down, it goes immediately to the share page and then can immediately share something.
Right, okay.
So if you fall asleep with it and it lands on your face, you can accidentally share videos to anyone on the channel.
Yeah, okay.
So I was like, I'm gonna get rid of that.
Yeah.
But I mean, there have been science communicators on the internet for a long time. People like Rebecca Watson and Hank and John Green. They do a really good job trying to talk about science and improving science knowledge from people. But there's also a load of grifters and people like your Andrew Tates or your Jordan Petersons who make videos promoting pseudoscience and nonsense, and those come out a lot more quickly, and they don't have to put any thought into it, they don't have to put any work into it, because you can just say whatever you want, and it'll be spread around by the algorithm. It kind of pollutes the algorithm. I don't really have a solution for that, except better content moderation that none of the
okay, so in a way, TikTok's not a brilliant forum because there's too much noise, there's too much misinformation.
I would say it's a lack of regulation around what is allowed to, or like what level of veracity is required for posts and things.
Is there any level of veracity required on TikTok?
No.
No, okay, so there's zero level of veracity. Okay, Margot?
As we were talking about extracting DNA from a strawberry, like yesterday I saw there's this, maybe you know him, he's like an online science guy. His name is Big Money, and he's like on everything. I wouldn't be surprised at all if he came to the Festival of Genomics in the coming years. But he does a lot of kind of, he speaks in a kind of I think he's from London. This kind of like urban kind of in an urban way, and he explains science, and he does a lot of experiments, which is very exciting. And he's got this deadpan delivery. You know who I'm talking about? Big Money, the black guy who's like, he's a science teacher in real life. But yesterday I saw that he did the strawberry extraction, the DNA extraction with Prince William. So Prince William is like the assistant, right? So he holds the bag and then put it out. And I think like whatever I think of the royal family, I thought it was like a cool thing because you're blending two things, and the science communication, the exciting part of it and the creative part of it is out there. And obviously Prince William gets a lot of great publicity, which I'm sure is good for him as well. And the other
Prince William needs a bit of publicity. We're talking about how to TikTokize. Is it worth TikTokizing your science when you've got all this noise that's on the platform as well? We heard about breaking it down to small bits, making it more discursive. I think are two things I'm really picking up from that. Go on, Matt.
Maybe we just need to outcompete all of the bad stuff, because it is very, very upsetting. Like I saw Joe. Maybe you want to share the final thought before.
No, go finish your point. Come on.
Well, it's a long point.
Is it a long one?
Yeah.
Is it another story?
No.
Okay, another story about vitamin B12. Okay, anyway, Molly, we'll come to you and then we'll find out what the story is.
I have a lot of thoughts about TikTok, but I don't have time.
Yeah.
But I have a fun B12 fact.
Yeah.
So B12 is the most expensive vitamin to produce, but it's not based on how much B12 you need to take. It's one of the most affordable.
Right, okay.
Because you only need like a microgram, like it's a very small amount that we need per day. So even though it's like one of the most expensive vitamins per gram, it's actually one of the most affordable.
Okay, cool. Well, thank you for that B12.
Can you overdose on that vitamin?
For vitamin B12, if you take more than you need, you can just pee it out. So it's not going to build up in your bloodstream or your tissues or anything. But there are anecdotal reports of acne if you have like a major overdose of B12. So you can't really overdose on it. You'll just pee out what you don't need. But yeah, most people should take a B12 supplement because it is like being depleted in like the food sources, especially like vegetarians and stuff like that.
Okay, and just want to, before you go, because that was a final thoughts, a final facts, but what about all the things we've been talking about, how to make the festival more fun, how to convey the passion of scientists in next year's festival, all these any final thoughts on that?
I think this should be available on a bigger scale. You guys should have a whole like section because this is really cool. This is like a panel discussion, but for anyone who wants to rock up, I think this is really awesome. So everyone should be privy to this conversation that we're all having. Okay, well, thanks very much. I will say that if you make it too big, sometimes it's, sometimes you lose that, but you lose, it's a bit like the TikTok thing, you get more noise and don't really find out about people like Daisy and, what's her name again? Florence.
And Florence. We don't find out about them if it gets too big. This size is a nice size, but where do we want to go with this conversation? Do we want to talk about this? You had something to say, didn't you, Matt? You had a story, another vitamin story. Eva. I
was going to say I don't use TikTok much, but I do like bite-size. I like YouTube Shorts. I like bites. In Instagram, the kind of various stories and that format. And so I think whether it's on social or not is not the main thing for me. The accessibility of being able to summarise some things. And you sort of said slightly provocatively that all the scientists should create a TikTok. Well, no, I don't think they should, but I think that they all should practice creating digestible, easily an easy communication that summarises the research, and I think that's actually quite a skill. And I think that however complex the research, if you're at a party, you can generally explain it to someone, and there should be an exercise. Like imagine you're at a party, explain your research, and there should be like mini abstracts of this sort.
Do you have to explain it to a drunk person, or is that making it too hard?
You can choose. You can choose what level you go at. If you have trouble summarising your research, you can you can even say a non-scientist who has got like a good grasp of stuff, or if you want to really, go down, there can be guidelines and stuff. But I think that helps everybody, not just lay people, but I think it also helps other scientists. We had someone here earlier who's a clinician who had a high level of medical knowledge, but didn't necessarily have an in for some of the more but might have been interested in that had they actually understood the abstract a bit better.
Okay, so it's actually vital for people of different fields to be able to understand each other. This summarising is not just for TikTok, it's actually a key skill, says Eva. You're nodding. Florence, what I mean, are you involved in science? Are you a science communicator?
I am. I'm a content producer. So I produce content from research papers.
Right.
So I think it's really, really important that researchers are able to communicate their work in a way that's accessible for people. And as you just said, actually people will miss out on things that they would actually be really interested in because it wasn't written in a way that they could understand. So I do think it's a really important skill.
I mean, when you talk to scientists and try to advise them how to communicate, what are the main sort of things that come up, or the main issues about how to summarise their work?
I think my tip would be, when you're explaining something or when you're writing something, the aim is to make the other person feel intelligent. So you want them to understand it, you want to use words that they're going to understand. And I think sometimes people will try and write in a way that makes them feel clever. They'll use long words or they'll convolute things because they feel smart writing it, and that's kind of not really the point of communication. So that's the thing I always say.
Okay, that's a big one. Make the other person to feel clever, or encourage the other person to feel clever. Margot?
No, I think I really agree with this point, because sometimes, like you have to ask yourself, who is this for? Is it to kind of make yourself feel good, or is it to actually communicate something? But I do think that this kind of workshop of how to kind of express yourself in a concise manner, it's a great skill, not just for science but also for the party, as you said. I think it's just absolutely crucial. So you may be a science person learning how to present your thesis in a TikTok video, but actually you are gaining a lot of social skills that can help you at the party, that can help you get a date or whatever, or get you to that next job. So definitely.
I mean, does it work with dating as well? Like,
maybe if you're a data analyst, I don't know.
Summarize who you are in a short sentence.
I think so. It doesn't even have to be like a pitch. Oh, I'm really good. It doesn't have to be about me. It could be, just something that I think this person would appreciate, whatever that is.
Okay.
And if I'm a geek, then obviously I'll have a lot of knowledge about a specific thing. It could be really boring.
Okay, so how do we actually summarize ourselves, not just the work we're doing, but who we are? Vital skills in a party, says Margot. I mean, I don't know if I've ever summarised myself in a party. Maybe that's where I'm going wrong.
I think it's also good for funding. It's good for communicating with other things. It's not just, it can be good for you without it necessarily just being those kind of outside of your profession sort of areas. I think it's really interesting that some of the best talks that I've been to have been the clearest ones. Not just because I haven't understood the others, because, I've understood at a certain level, but they haven't engaged me in that same way. And I think actually it's a sign of true intelligence to be able to speak at a level where anyone can understand what you're saying. And by understand what you're saying, I'm not saying that you have to understand the intricacies of every experiment and every thing that's been shown, but you're following along on the main points, and that is a skill. So I think we were talking earlier about different crossovers of science and art, but I think that science communication should be something that is on every university
could actually lead to
course.
crossovers with science and other scientists.
It can use to crossovers, synergies, funding applications, being able to reach communities other than funders who might support your work in other ways. So, I mean, really, I'm sure it's been documented elsewhere, but the benefits are massive.
Yeah, and manifolds. Finn, you've not said much for a while. Any thoughts on this? And this person's going to come and join us. Do you have problems TikTokizing your work, turning it into bite-sized social media chats? Give us a final thought, Florence and Daisy, before you go.
we need to make communications more accessible, and we need to make sure that it's reaching the right audiences and being communicated in a lay way. So like translating that complex scientific stuff into lay.
Is there any like magic formula for doing that?
Just like imagine you're talking to your mum, or like.
Yeah.
That's what I think.
Yeah. Okay. Yeah. I do that a lot, actually, imagining that I'm talking to my mum. Sometimes I do actually talk to my mum. There's a question
Just
for you.
Just a quick question. Do you use a lot of do you use any jokes? Is it appropriate to use jokes or gags in explaining science, or is that just too familiar, counterproductive? Because I really like jokes, but it's hard to make them in a science context.
I mean, there was a lot of complaints yesterday. There were not enough gags on Talkaoke yesterday, and somebody put in a complaint about that. But can you use humour?
Well, you could probably use some analogies, but I think because if you have a certain brand, the way that you want to come across, maybe it's not appropriate for a lot of for some companies.
Okay, so it kind of depends what you're communicating.
Exactly. Yeah.
Florence, final thought.
Agreed.
What are you thinking? It's fine. It's fine. It could be anything.
I was just thinking about the point about using humour. I think it depends. I think you can do it in a subtle way. We use analogies, we use metaphors so people can kind of understand things, that language they're familiar with. But I think if you have a lot of, like, if you need people to have trust in you, then maybe like making jokes, I don't know, maybe that's not the best thing.
Yeah.
But
okay.
Yeah.
So it can work, but sometimes it can also fall flat as well. Yeah. So it's a risky tool to use, humour, but. You know, I'm going to take the risk and try to. Well, if the opportunity comes, I'm going to try and throw some gags out there. We've got a new face. Thank you, Daisy and Florence. We've got Finn, we've got Matt, we've got Eva, we've got Margot. Tell us your name.
Venita.
Venita and Mikey. At the moment, we were talking about how do we make this festival more fun for next year. Then we talked about the passion of scientists and non-scientists, what makes them actually do the things they do. Then we talked about doing some live experiments in the festival itself. Then we somehow moved on to talking about how to communicate what you do more. Oh yeah, we're talking about graphs because a lot of people felt there should be some kind of competition for the best science images. And then we talked about collaborating with artists, and then we talked about communicating what you do on TikTok and other social media platforms. And then just at that time, and that's some of the serendipity of Talkaoke, we had some social media science communicators. They're the ones who just left, giving us some insights into how to communicate your science on social media, and that's where we are right now.
But I'm interested in this point of kind of maybe not using humour, because maybe that would undermine how serious you are in what you do. And yet you've got Brian Cox performing later tonight or speaking tonight. That hasn't always been his main thing, science, right? So he was a musician, from what I understand. And somehow, is that okay? Is music okay? Is that refined enough? But a stand-up comedian come scientist wouldn't work? I'm just curious, like what is the threat?
You can do multiple careers, you can be a musician and a scientist.
Yeah, but I think maybe, I don't know, I just feel like it's such a shame if people feel like they can't make a joke about what they're working on, like that's going to undermine you. I don't know.
Yeah, okay.
I think so. With each, if you're not sort of communicating just for yourself, you're communicating very often like a science communicator will be communicating for the company or the health body that they're communicating for. So it's not the same, obviously,
so
as
you
the
communicate
way
for the institution, really.
Yeah, and then you will have a tone of voice document about what is appropriate and what isn't appropriate, which doesn't, and you know, there'll be slider scales, you know, between irreverent and serious, etc. And I think that you do have to be incredibly careful with humour, because if you're communicating with a lot of the public, they want, you know, it's okay to do it like, you know, hey, we've put a Christmas hat on, you know, microscope or something, just, you know. But if you do so little kind of things like that, you know, fine. But I don't know if that's what you're referring to. Anything that's more deeply into the humour is almost wrong in relation to that context, because the people who will be following, the people who will be getting involved will be patient groups, patient bodies. You know, there might be someone with a serious ill child, and so some types of humour just won't feel right for, you know, we're going to say the brand without it sounding too corporate. if you're representing your employer, then there is a structure within which you must adhere and communicate.
Okay, so humour sometimes is a bit trickier when you're doing very, very serious subjects, Matt.
Two points on that. Once when I was presenting, I included the picture of my cat because I thought it would be funny. No one got it. Literally, no one. I moved on. No one got it. No one said, "Oh, well, I didn't hear anyone."
Was it in relation to this thing about making your cats fluorescent that you were talking about earlier on?
Well, maybe it's because no one likes black cats, and I have a black cat, so it's their fault,
I mean,
not
do you
my
feel
fault.
like it was worth trying just to mix things up a little bit with a bit of humour?
Yeah, I try, but I was presenting to scientists, and scientists are boring.
Okay.
So
go on, Vanita. Yeah.
I was
Grab
just,
the mic. Vanita.
I was just going to say that humour has its place, but humour is very culturally appropriate.
Right.
So what you might think is funny is not necessarily funny to an Indian or a Egyptian, or so that's why humour is limited. But in terms of communication, there are lots of perspectives, like what the company wants to send out, what the 5-year-old needs to know, what the 10-year-old needs to know, what the elderly. But I read some of the stories there, and one mum had written, the doctor had asked her what had changed since the last time he saw her. And it was just beautifully how she put her perspective that, she was at a wedding and she felt really emotional, mum wasn't there, and the child that's affected with the genetic condition held mum's hand. And I thought, something like that can really touch you. So I think communication has to grab whether it's your spirituality, whether it's just your understanding. For me, graphs I just don't like graphs. So I like communication in different ways.
I mean, it's interesting because someone earlier on said they went to a talk, one of the first talks here this morning. I wasn't here yet, but they said they felt very emotional and it affected them because they were. Someone was talking about a relative that wasn't there anymore, but they thought that was actually very important, and it gave the work that they were doing some gravitas and some meaning.
But I just wonder, because I think emotional stories we can all relate to, but I think they're the very things that are being appropriated. For me, that's the risk, because you see people, making misinformation and publishing misinformation online. It's like, oh yeah, my mother, and there's so many scams going around. I'm from Poland. There's it's not uncommon for people to get scammed because, oh yeah, your daughter's in an accident, you have to transfer the money, whatever. So I think emotions are just so strained.
Yeah.
So I think humor is almost a scientific art in itself, because I agree with you, not everything is going to be funny for everyone. But I'm not saying, oh, should we just develop a generic sense of humor so everyone can get on board with, but maybe that's already happening anyway. But I do think that to get the tone right it's a real skill. It's almost like a science in itself.
Yeah,
but it is
science of science communication.
But you just don't know how it's going to land. I mean, reputation, of course, but maybe, I don't know what I'm saying. I'm just saying those things. I think emotions are quite easy to exploit as well.
Okay, so emotions can be manipulated as well, yeah. I just want to bring in Finn, because you've not said anything for a while. Any views on any of this? Do you feel like humour, emotion are good tools for science communication?
So, in terms of humour, I think of the American science communicator Bill Nye, not Bill Nye, the actor. He did a lot of science programs for kids, and he made them very fun for kids. And a lot of people said that they were sort of people's entryway into science was watching Bill Nye's shows, and that has lots of jokes in it. But that's aimed, as people were saying, you got to aim it to your audience. It's aimed for kids, so it keeps them entertained. When I'm giving talks, I always want to add jokes in, but there's always the chance that you'll make a joke that just doesn't land. Especially when you're as unfunny as I am. But I do find it, when I'm giving talks to people I know, so departmental seminars, and I know people in the audience, you can say things that just everyone is going to kind of get as a relevant thing for that, which helps keep people engaged. My mum used to say she'd always swear about 20 minutes into a talk she was giving, because she would know that everyone's slowly starting to lose interest, and then they suddenly hear "fuck," and everyone like, what? So I don't do that, because I'd absolutely get fired. But I think just having those little joke breaks and things in, if you can put them in the right places.
So an expletive halfway through, I think it's okay, it might be controversial. Have you ever sworn in a talk? Come and join. You can sit and listen here, and it just makes the whole thing, seem a bit more, bit more of an audience, a bit more popular. But you don't have to say anything if you don't.
You are not going to make me talk.
I'm just going to find out your name. Just find out your name.
Burcu.
What's it?
Burcu.
Burcu. Okay, so we have got Finn, we have got Matt, we have got Vanita, we've got Eva, we've got Margot. We didn't get your name. Roisin, and I'm Mikey. At the moment, we've gone down quite a long road where we started off talking about what would make the Festival of Genomics more fun next year, and there's lots of ideas: live DNA extraction from strawberries, strawberry daiquiris, a bit of cocktails going on. Art competition, who makes the best graph, and then we talked about, okay, do we need to work more with artists to get to do better talking and better performance as scientists, but also better aesthetics. Then we went down that road, and then we talked about social media, the TikTok science phenomenon, is it good or bad? And then we're kind of sort of talking about stuff around that now, about humour, is that a good way to deliver your message, or emotion, talking about real stuff and the real seriousness of some of the research that people are doing. So that's where we are right now. And then we've just had this idea from Finn that your mum uses, which is a little expletive halfway through the talk, just to wake people up. And I think as a poet that actually works well, to just the odd peppering of expletive gets your audience definitely listening.
Just a little spin on that, because I think to replace the expletive, we can, for example, use a fart. You see, farts are funny. Everyone loves a fart.
There was a fart sound effect on here, but it got taken off, actually.
So no smell, just the sound. Halfway through the talk, the funny sound. Because children, like I have a 2-year-old niece. She loves the sound of a fart.
That's why we had to remove this fart sound effect, because the kids would keep on pressing it when we do a kids gig. But maybe we could use it actually for the Festival of Genomics. We could bring that one back.
Definitely.
Yeah, okay. Yep.
This reminds me of a TikTok, and I'm not on TikTok, sorry, I think I saw it on Instagram, it's a trend called Guess My Fart. Has anybody heard of this? Obviously, you're an expert. Basically, the way this works, like, for example, there'll be a couple, or like 2 people together, and somebody's like about to fart, and they say to this person, guess my fart, and they have to just imitate the sound that they think is going to come out. And if it's right, it's really funny. If it's not right, it's maybe less funny. Obviously, you don't get the smell, but you get the sound, and apparently a lot of people do it. I haven't done it, but I'm thinking.
I mean, could we do something like that for next year's Festival of Genomics? Would people participate? I think,
even better, let's do microbiome sequencing and then guess the fart.
I mean, can you actually determine somebody's fart from their genome? Has anyone done any research into this? Well, there was a guy, actually, we started off at the very beginning with a guy talking about fecal transplants and how somebody's microbiome was so important to how they would actually, if they got these degenerative diseases or not. So maybe we could incorporate that into guessing the fart. I don't know. Anyway, where am I going with this? I don't know. We've gone down a little bit of a black hole here. Roisin, help me out. Any thoughts?
To jump back to presentations, for a long time, I've not been doing presentations for a hugely long time, but I very much peppered in jokes and stuff, just because I could feel people just waning and leaving the room spiritually. But I recently watched a talk at a
like a small meeting that I was at, and the woman spoke so confidently, so sort of, she put the room at ease. I had no understanding of her.
It was a presence that she had, yeah.
Yeah, and I had no understanding of her subject area, but I left the talk, I knew what she'd spoken about, and the reason I knew it was so good was a couple of hours later my partner picked me up in the car and I was able to explain to him what she had done.
Right.
And she didn't use any sort of humour, but it was just the way that she was, and I now really want to do that. Like I don't want to have to rely on like pulling a room back by making a joke. Like the way that she presented was so
you
good.
don't have to make a joke or fart or swear. It's just about having presence. You're a performer, Margot, you mentioned earlier.
Yeah.
Have you got anything to say about presence? Or?
I do. I think presence is very important, but I still think that you can use humor. But I think for me, it's also about the voice, how you use your voice. Because I've got, like, I recently went to a talk, which was it was an artist talk. It was an exhibition, and there were a lady was making an interview with two artists who were exhibiting, and she was asking really great questions. Like, I was like, oh, she's really knowledgeable. Her voice was so boring. I had to leave. I had to wait until it's finished, because I'm just so sensitive to people being really boring. And I think, I don't know if it was her accent or whether just lack of awareness. They can really explore the range. Like, we had somebody who's a voice therapist there earlier, and you could really hear in her voice she's exploring different ranges as that, and that is the way to keep people hooked in. I've got a friend from Manchester. He speaks like that all the time, and I'm not doing it wrong because I can't actually do accents, but I've noticed that I just switch off when he talks to me because I can't. Very intelligent guy.
Is it about varying the voice?
It's about varying the voice, I would say. And then you don't have to be funny, but you catch people's attention, and I think that's why fart sound is just so good, because it's just very different.
Okay, so you're actually just exploring different sounds. It's like, it's almost like the difference between a repetitive kind of techno beat that we know the physicists like to something that's a bit more, a bit more
I know you can't sing, but
explorative. Ever.
I have to go, so I'll do my final thoughts and try and link some of this up as well. Just to say, maybe we should get people, like, there's a kind of like an assessment, and people who do not have enough engagement in their style, etc., should spend a year reading stories to children in schools as kind of, do a DPS, get the voluntary thing. Because when you read stories to children without kind of patronising adults, you do have to, you do the eye contact if you can, and you can learn that if you're on the spectrum, so that you have trouble with it, but just in order to you don't have to actually look at anyone, you have to vary your voice. You can learn how to be more engaging, and I think a bit earlier we were talking about communication skills being part of science degrees, being optional compulsory parts of it. So there are ways and things you can do, but
so
my jokey polemic.
Children.
Yeah, well, I found 5 years of reading stories means that now when I read any text, although I would not advise anyone to do a presentation that was just reading a text anyway, but you learn to make your voice, I think, quite a lot more expressive, hopefully. Otherwise you get your kids to sleep quite quickly.
Okay, so you might want to make it more boring if you just want to get those kids to sleep, but if you don't want the kids to sleep,
apparently there's a book that was a big hit about 5 years ago, because it was incredible. It was a book for children that was written to be boring,
right?
So that actually, I don't know if anyone remembered it. It was like, it had a weird.
Was it called a meta-analysis of genes or anything like that?
Probably. I think there's a few candidates on the program today, but apparently it was meant to be very repetitive and very boring, which I think is a real shame. So that's not what story time's about.
Okay, all right. So story time should be fun. Thank you for that. We're going to hit you with a round of applause. Okay, Roisin, you wanted to say something? Yep.
I was just really liking to build on that point of not only is it about you being engaging as a speaker, but I think as an audience you hold some sort of accountability for like, I'm a big nodder in talks, so I nod along a lot. Because I find that very helpful when I'm up there, that someone's like, yeah, I get what you're saying, keep going. So I think as an audience member, you also have a responsibility.
Okay, so you need to somehow physically engage by nodding, or otherwise you're nodding as well, Burju. But I did kind of make a pact with you that I wouldn't talk to you about anything. However, don't feel like that. You know, if you have got something to say, grab the mic. I don't know if I'm just going to offer it to you now.
About the ideas, I was thinking of instead of guess the part thingy.
Yeah.
What about guess my specialty?
Yeah. Okay.
Or guess.
We can
what
play that
I'm
now because you've got your lanyard covered. We could guess your speciality. Yeah. Okay. So we've got Burju. We know nothing about her. Can we guess what her speciality is? And we're going to go around the table on this one. Finn, have you got any thoughts? Can you ask questions, or is it just a straight guess? Just a straight guess?
I don't know. You
No.
are the host.
Okay, all right.
Invented this game. You have to make
No, you came up with it. I'm just going to say, let's just go for a straight guess straight away, just on the basis of appearance.
I'm going to go safe and say something to do with genomics and biodata.
Genomics and biodata. I think you've got to be a bit more specific than that.
I don't think I do.
No, you don't. Okay, all right. You're right. It's your Talkaoke. It's up to you. It's this is your forums.
You're a chemist. You've got something with chemistry.
Okay, chemist. Yep.
Biochemist.
Biochemist. Okay.
I was going to say studying science, MSc, something.
Yeah, student. Yeah, okay. All right, Roisin.
I love that.
Yeah.
Maybe a genetic counsellor.
Genetic
I don't know.
counsellor.
You seem very friendly and approachable. So.
Okay. All right. Can you tell? Or should we go around and ask questions now? And who's the most right out of all those people?
I think these bits.
Okay, Benito said you're a student.
I'm not a student anymore.
Yeah, okay.
But I have done genetic counseling a little bit.
Okay, all right, so...
But chemistry, I am nowhere near it.
Okay, so... All right, so, I mean, should we do another round to get it more specific? Okay, we know it's something about genetic counseling. Is that enough, or do we want to hone down this a little bit more?
It's hard. I mean, it's hard to guess because I don't have a specific specialty right now.
Okay, right.
I have studied molecular biology and genetics.
Right.
Then I have done, for a while, I was a laboratory coordinator in a genetic diagnosis center. That's why I was interacting with patients, giving them genetic counseling. At the same time, managing my people, colleagues, to do their tests. Afterwards, I went back to R&D, and I was developing some ingest panels.
Okay, so that would be very hard to guess, though. I think they've done very well. So let's have a round of applause for everyone. You got a little bit of it as well, I think, Finn.
Yeah.
Okay, we kind of semi-guessed what you do. Where do we go from here? This is your forum. It's kind of up to you.
Say about for next year. I love the idea of. See, you're very dramatic and you've got good speaking. I think there should be a little bit of a theatre show.
Yeah.
Representing, I don't know, DNA or variants, or I like the idea of something interactive.
Okay, some kind of theatre show.
More of a play show, yeah.
Okay, it could be some kind of immersive theatre. We've got. What do people think about that? What would the story be if we were going to make a theatre show?
I mean, because we sometimes do this format which is called Segue, which is to get people, it's not like showing people something that, here's the one I made earlier. It's more like setting up a film set and then getting people to step in and then just devise the story, make the costume. I mean, can you imagine what costumes would be like? Like, because I can't, because I'm not a scientist, but I imagine it would be pretty bonkers, right? Just bring a lot of like foamy materials, I don't know, like fabrics.
We can maybe do some protein folding in the costumes.
Yeah, just like fold it in, fold up your costume. So so we could actually make a film on day one, on day two, just screen it at the end of the day. Wouldn't that be amazing? And then, because I don't know what,
we'll pitch it to
because people would have ideas of how to, and all jokes go if we want to, characters can change. It would be so much fun. I want to do that. Thank you for this.
Okay, thank you. We've got that recorded. Would people take part in a theatre production? Finn, would you get involved?
I like making a fool of myself, so probably would.
You would,
yeah. But I think part of the problem is a lot of people who get into science tend to be a bit more on the shy side, or at least in my experience. I know I certainly am.
Yeah.
I mean, even wearing a mask, I don't think helps people who feel that kind of way. And I wonder if by doing things that are big and explosive like that, and showing a film to everyone at the end, would make people less likely to engage in that kind of open expression of things, because it's very personal to do. I'm a musician. I do a bit of writing. I run D&D and things like that. So I'm not unaware of the expressive side.
I mean, you're probably not the only musician here. Would you get involved in some kind of jam or some kind of... Earlier on we were talking about not DNA sequencing, but beat sequencing. Could we actually exploit some of the creative potential of some of the musicians that are inevitably here to actually do some kind of jam or mixing session? And we're not talking about mixing different genotypes, we're talking about mixing different tracks.
Yeah. I mean, like I always love playing with other musicians, but I don't see how that would link with a scientific thing. It's very difficult to create that link between those two things. Often, we were discussing whether you can do science, art, and things. Art, to me, is not the process of producing something. It is like the process of taking emotions or feelings or thoughts or ideas and presenting them in an artistic way that can be interpreted by others, which isn't quite the same as what you want to do in science presentation, because what you want to do in science presentation is present your data in a way that other people can pick up on what you've done, pick up on any flaws that you may have, or gaps there might be they could offer you help on, and also suggest what you think, but also allow them to come to their own interpretation. So it's kind of like art in that you present something that has your ideas behind it and let other people make their interpretations of it. But art is quite dispersed in terms of facts, especially as you move away from realism and things.
So the issue is really about the data, the hard data, as opposed to the kind of soft data of art, or the...
It's not the difference between hard and soft, but the difference in science, one of the things we're trying to do is take data and present that factually, and then make our interpretations based on what we see. Whereas art, I feel, is a slightly different aim. You can take things from one for the other, but it's definitely a very different discipline. Sorry, I'm taking too long.
Roisin,
I slightly disagree with you, because I don't know if you were here yesterday, and we had the talk by Adam Rutherford on basically a bit of racism and subjectivity in terms of teaching genetics, and the first thing we were taught was like the inheritance patterns of, let's say, blue eyes or brown eyes, but lots of factors come into you receiving brown eyes or blue eyes. It's not the one
genetic story that we were told at school.
So I don't think genomics, and I don't think science is black and white, but I think that's what we're led to believe, and I think we need to break away from that.
It's almost like the myth of this kind of neutral data, the neutral facts, is a little bit of a myth as far as you're concerned. Okay, we've got a new person at the table. You've walked into a talk show. This is Talkaoke pop-up talk show, where we talk about whatever people want to talk about. We have got Margot, we have got Matt, we have got Vanita, we've got Roisin, we've got Burju, we've got Finn. I'm Mikey. Tell us your name.
I'm Edward.
Edward, welcome. And come, this person maybe is going to join us, maybe not. And this is a really interesting point. So Finn has come and said, science is about, you didn't like the hard and soft data, but about facts and presenting facts. But Vanita has said sometimes what is presented as facts has actually got some kind of bias or some kind of story behind it, and it's a bit of a myth of this kind of, this neutrality, this objective data. So that's where we are right now. Finn, you had something to say about Roisin. Do you have something? It's moved on now. Okay, you want to follow this up? Yeah.
No, but I'm just thinking it's an interesting question. What is art for? Because if I'm making a theatre piece, shall we say, in response to a paper that you wrote, that's one way of using art. But, like for instance in philosophy, or even Isaac Newton with his apple, what has got apple to do with anything? And yet, it's got everything to do with it. It's more about, for me, using art is pushing the ideas forward, and it's not a linear way, but it may just make sense for you personally because you're working on XYZ, and this imagery that the artist or the poem or the visual, the film or whatever, it kind of made sense for you. You're gonna go away, think about it, and that's what I think is good about involving art. It's not function. It doesn't have to perform a specific function like a scientific paper. That can do its own job.
Okay, so sometimes the ideas come not in a straightforward, direct, convergent way, but they're a little bit more sideways. We've got a new face at the table. Tell us your name.
Hi, I don't know what's going on.
What's your name?
My name is Marta.
Marta. That's it. That's all we need at this point. We have got Finn, we have got Edward, we've got Burju, we've got Roisin, we've got Vanita, we've got Matt, we've got Margot, and I'm Mikey. At the moment, we're kind of talking about is science really just kind of objective facts presented without any kind of cultural background or any sort of personality of that person that's presenting it? Or is that like more of an aspiration, or is it just not the case? And we heard it just now about the kind of myth of genetic eye colour, that it's down to some very simple single genes, but actually it turns out that's just not true. And that was just something that made up possibly in a little bit of a kind of culturally biased or racist way to explain something oversimplistically, and that's an example of where science isn't as objective as maybe we think it is. So that's where we are right now. Edward, come in a bit as well. You're a bit far away there. Yeah.
Well, I mean, the whole point of science is to use the data you have. The more data you get, the closer you are to the truth, which then you can falsify. If you can't falsify, then it's more likely to be the truth, but it's never definitive that it is the truth.
Right.
So the more we progress, the more things will get falsified and new things will come about.
So, I mean, what you're saying really, Edward, is there's a process of science. It's about presenting data, presenting an explanation for that data that is potentially falsifiable. And that is a process which is very scientific and maybe does not apply to artistic practice or other forms of culture.
Exactly.
Okay, all right, that's I mean, has that kind of nailed it? Do people feel happy with that? Yeah.
So sorry, I think that the data you've got is one perspective, and you're right. You know, that's got to be replicable, but it doesn't explore everything. For example, I mean, you might shoot me dead now, but the talk by Tim Spector and Zoe. His application and the microbiome being so important in terms of you know, our overall health. Microbiomes associated with everything now, autism and brain health and happiness, but I do think that a microbiome is really important. But I think there are some other factors that we in science you can't capture, and I think just because we can't capture it, it's not research. So, for example, the microbiome is important, but what's important is your state of mind and how you feel about the food, whether you feel guilty or you feel that that's, you know, nice, or also, another thing that we in sort of our Indian culture, Ayurveda, it's very important in terms of eating, is the time of day. So, for example, your main meal, they say, should be when the sun is at its most, prominent, midday. So your digestion, so our body clock is aligned to the clock.
Yeah.
And so we would digest more easily. We can absorb that heavy meal at that time, but later on it's very difficult. Now I know that in our Western world our timings are different, our working hours, but you see that has an impact on.
It's a different form of knowledge, but also there's something about experience as well. The experience of eating and enjoying food, as well as just the pure kind of chemical content of that food or biological content of that food.
And food, we talk about eating fresh and local. So that's really important because, like in Ayurveda, foods are termed as sattvic, which means that, so for example, if you're having an apple, when it's fallen off the tree, it's nice and fresh, and it has its own kind of this life force energy.
I just want to remind Roisin, you've been to the table before. We need a final thought from you before you go.
Roisin, don't go away.
But
don't
that
get
means
bored.
that's a little bit of pressure of you, Vanita, just to finish this point.
Sorry. So I'm just saying that everything we eat has a life force, when I talk about that, I mean things like the fruits and vegetables. So they also matter in terms of when you eat them. So if it's like dead and wrinkled, you know, that's going to give you that terrible life force. But sorry, Roisin.
So when it comes to vegetables, it's better to eat vegetables that are alive. Eat them alive. Don't eat them dead. Yeah, I'm going to take that from Roisin. Final thought.
I never thought I was a creative person, and I think that, you know, when you were assessed at art in school, I would never understood how you could be good or bad at art, but I was very much graded badly at art, and I think that is one of the reasons I got into science. But since then, I've become creative in my own time, and I still struggle to see that as creativity, but I think we also need to see the work that we do as scientists as creative as well.
Okay, that's a really good... I'm glad I stopped you for that final thought. Okay, so there's definitely a creative element, but you're talking about data and you're talking about process. You're not necessarily denying creativity, you're just saying it's a different process. Edward, you're off as well. Give us a final thought.
I'm very confused in this talk. I don't know really what's going on.
Well, in itself, that's good. In itself, that's good. But I guess, if you want, I can sort of like help you narrow down the final thought. So we're talking about, in a way, different processes: the scientific process, the artistic process. Are they as separate as some scientists would want to make them? Is there more of an overlap? Okay, you've outlined a process that is a kind of scientific method, but what about this point that Roisin's made about creativity? How creative are scientists, and how can they best tap into their creativity? Maybe that would be a good final thought.
Yeah, it's quite a thought.
Yeah.
Quite a thought. So,
I mean, do you feel like you're tapped into your creativity as a scientist?
I'm not sure.
Okay, maybe that's food for thought. Yeah. So thank you, Edward. Yes.
Hi, I just wanted to add to this. I read a study.
Yeah.
I didn't read it throughoutly, but I just read it was published, so I would assume there's some truth to it.
Yeah.
About how creativity is linked to awe, like the emotion, awe for the natural world or anything like that. So I do think a lot of scientists, like, there is some correlation, and a lot of scientists are actually creative. They just don't see it as being creative. Because of like the STEM stereotypes and stuff like that, I think they kind of are put into boxes. But
so they're not necessarily celebrating their creativity.
Yeah, exactly.
Like
Whereas I think actually the whole premise of science is really creative. Yeah. I don't know.
So that's a good one. Or we're talking about awe. Is that what we talked earlier on about what got people into science? Maybe awe is a fact. We didn't talk about awe before. And it's a different one to talk about because it's a word with no consonants, really, when pronounced in English, awe. But anyway, I think it's a good one to throw in there, awe. And is that a factor when it comes to the passion and creativity of scientists?
I mean, this is an excellent point. I also want to pick up the Ayurvedic point because it's not a subject I know a lot about, but I'm very curious, and I'm definitely going to go away and read up about it. But in terms of the awe, I think it's exactly right. I think facts kind of kill the awe because, for instance, by learning more about history or philosophy, because philosophy, for example, it can be so untangible. It's like, oh yeah, it's just somebody thought this, and suddenly the whole paradigm is shifted, and but it happens incrementally. But I think when you realize that of history, oh yeah, there was this battle or whatever. Fast forward to 300 years, we live in this kind of world, not that kind of world. These small things do have impact, and I've noticed that in recent times that somebody would be telling me about, some kind of historical fact, and I'll be like, isn't that amazing? I just find myself going, wow, that's amazing. Somebody actually died for this thing, or they actually decided, no, I'm going to do this instead of that. I think I definitely had my awe reinstated, but not by facts.
Is awe important to you?
Very much.
Okay, interesting. So, thank you for that, Marta.
It's not either or. It's
not an either or.
All the way.
Yeah. Okay, where are we going to take this? I don't know. I feel like with the awe thing, it is a bit of a final thought. It might be time just to have a little break. I've been doing this a while now, but let's just get a final thought from everyone. In a way, we could be talking about what inspires you, but also creativity as a scientist. Also bringing in different and I'm going to use this word unashamedly, different knowledges that are not necessarily part of the mainstream scientific dogma. How important are those things in inspiring us and actually moving us forward? So, who wants to go first on a final thought? I'm going to get to you, Finn, because you always got something to say. So,
I'm not sure.
What inspires you?
What inspires me
as a scientist? How did you get into this in the first place?
So, I mean, I was going to go down a creative route and trigger warning for everyone on the table for mental health issues. Got very, very suicidal and decided to do something that would provide value. I now realise that actually creative arts and things are incredibly valuable. I have a lot of friends who work in the arts, and that's really, really valuable and useful. But yes, that was what got me into science, was feeling like I had to give back.
You had to give something. You wanted to give something to the world.
I had to earn my place in existence, which is a terrible reason to do anything.
Do you think so?
Yeah. Yes. Yeah. No one has to earn their right to exist.
Yeah.
Maybe Elon Musk should do something worthwhile for a change. But no, you shouldn't do something because you feel you need to earn your right to exist on the world. You should.
But I mean, what about this question about contributing? Because I think we get this a lot, even on Talkaoke, because in a way it's about contributing to the discussion and contributing to the kind of community that the sort of, just spontaneous community that we create here. And people love to sort of throw things in, throw in ideas, and that is a lot about what drives this as a practice, or what drives people. So I don't think it's that kind of crazy a thing to feel like you need to contribute, that you want to give something to the world.
It's a difference between wanting to contribute and feeling you need to contribute, because everyone contributes to the world just by being in it.
So it's more like more of a sense of desire and less of an obligation.
Yeah, I would like to see a world where everyone understands that they're irreplaceable and priceless. And they have something to contribute, whether they feel they do or they don't.
That's a great final thought. Yeah.
No, but I think one of the really great benefits, not that art needs to have a list of benefits, but one of the really great things about art for me is that it taught me or showed me that your value is not reliant on achievement, like an upward achievement. Or I'm going to be, I'll achieve this qualification, this job, this amount of money, this kind of status. Art, it really kind of blurs it, just destroys the vertical? Hierarchy of, of life and what achievement.
Is it more of a process, or is it?
I don't know. It's like everything. It's the process. It's just like you can see that you can gain a lot of things which maybe other people don't value, or they don't know they value. They take them for granted. And to kind of stand your ground and say I'm an artist and this is what I do because I think this is worthwhile. Yeah, okay, maybe somebody won't quit their corporate job on the day of that conversation. Maybe Elon Musk won't become a good person overnight, but I think you do have impact on people. Is that kind of the butterfly wings somewhere at some point, as a cumulative effect, they will cause a bigger change than just so that's how I feel about art. So this is an excellent point I thought you made, and I just wanted to add. That's my final thought. Thank you.
Your final thought. Okay, we're getting final thoughts now. Who wants to go next on the final thought? I'm not going to say Matt, because you only just got back here. You've no idea what we're talking about, so I'm going to give you a couple of minutes, but let's get some final thoughts.
I have a cute story about how I get into science. When I was little, I was hating all the vegetables and stuff. They were either too, I don't know, spicy, or some of them were prickly, tasty, something like that. And there was in my home country, I'm from Turkey, there was a kid magazine for science for kids kind of stuff. And in there I have read about genetics, and I have read about how people are changing plants and stuff to make them in a different shape, etc. That's why I got into science. Afterwards, I realized that I had mild allergies for some of the vegetables. That's why I was experiencing the
Science explained why you didn't like vegetables.
Yeah. Okay. I didn't study plants, but I found my answer.
Have you ever genetically modified a vegetable?
No.
No. Would you like to, or have you moved on from that?
I think I moved on from that.
Yeah, okay. but a great story, how you
Thank
got
you
into
very much.
science. And yes, anything else you'd like to add as a final thought?
Thank you very much. You're a great host.
Thank you.
Well, teşekkür ederiz, we could say. Thank you for your contribution. Marta, Benita, who wants to go next? Final thought? Marta, have you got a final thought?
Sure. I don't know. All of this talk about doing what you want and believing in it, it reminds me of one of the greatest cancer researchers, I think. I think his name was Weinberg.
Yeah.
I'm not a cancer biologist, so I don't know.
Yeah.
But basically, he published a paper, Hallmarks of Cancer, very, very famous paper. But back in, I think, 70s, he attended a conference, and literally people, like he was very young, and a lot of people told him, like, do not think of cancer as a molecular disease, whereas nowadays, that's the only, like, direction we're going in, which is, yeah, I think, and we've seen this like mRNA vaccines and RNA in general. I think just believing in what you're doing is very, very important.
Yeah.
But yeah, cool.
And sometimes going against the grain as well, and actually standing up for what you believe in.
Yeah, exactly. Yeah.
Okay, and for you, is that mRNA? Is that your
No, I
thing?
just really like RNA. So I'm a big believer in RNA in general.
Okay, so with not enough light is shone on RNA, we're just so too DNA focused in this festival. Do we need a separate RNA festival, or is it just about changing the paradigm for genomics and talking more about RNA?
That's an interesting thought. I think it's a bit more about changing the whole.
Yeah.
I mean, it feels a bit tricky to say.
Don't worry, we can. It's my job to support you as an RNA person amongst all these DNA people.
Yeah. So
exactly.
I mean, it's not about changing the dogma completely, but maybe looking at how RNA influences DNA a bit more in depth, and maybe what we can do with it. Because, for example, changing DNA is pretty permanent, right? Maybe there's stuff we can do with RNA less permanently. I don't know.
Okay. All right, I'm going to hit the... Okay, Benita.
I just wanted to say that what I really appreciate about everybody, you're all scientists, how you came into it because of things that you'd experienced, and now you're putting everything into it, and your work on RNA and its importance, and Urji, your likings for vegetables or not liking, but that to me is the passion and driver, and to me that's really important. So all the it's
what
your
about you,
motivation.
Benita? How did you get into this?
Well, I read a book about Florence Nightingale, a ladybird book, and so I always wanted to be a nurse, so I started off as a nurse,
Right. Oh,
and
wow.
I've really enjoyed my career every day, I've really enjoyed. So I started in nursing and then moved into kind of health visiting and sickle cell thalassemia counselling and genetic counselling.
Wow, okay, thank you for that. I'm going to hit you with a round of applause. Matt, final thought?
I was just thinking, my capitalist hat is coming on. How about we just make genetically modified peanuts that just don't allergize people?
Yeah.
So, just eliminating the problem altogether. Use genomics, use the DNA.
Yeah. Okay.
So that's my final thought. I want to make money.
Okay, all right. Okay, we're going to leave it there. Is there a final thought?
If you're gonna modify nuts, don't do it on peanuts. They're like the most boring nut. Just do it on like the other ones, like Brazil nut. That makes people quite sick, but it's a very tasty nut.
Okay, so you think there's probably more mileage in the nut, the hypoallergenic nuts market than just peanuts, and we're going to leave it there. This has been Talkaoke. I think we're supposed to start up again in about 5 minutes anyway, but we've over-talked. But thanks everyone, it's been lovely to meet you all, and we'll be back very shortly.
The second session. Come have a seat. Observing is, Good question.
Oh, it's under here.
Everything going. Yeah. It's because. Okay. Screw it. Let's stick on our guards. Can you do it?
You got to turn on channel 1.
Turn on.
Channel 1. No, not that one.
That one?
Give us a bit more.
More?
Yeah,
now?
yeah.
Amazing. And
The one above that is on the at 12 o'clock.
this one?
Yeah.
At 12 o'clock. Yeah, it's at 12 o'clock.
Yeah, good.
Awesome. Hi, this is Talkaoke, a pop-up talk show where we talk about everything and anything. Please come and join us.
No, it's been logically tested, it's safe, you're fine.
We promise.
Yeah. We do.
So my name is Matt. I'll be hosting this sub-session of a Talkaoke. What are we talking about, Mikey?
I don't know. Maybe turn up channel 2 as well.
No?
I don't know.
You don't know?
It could be anything. There is a request to talk about. I believe we're kicking it off talking about equity and diversity.
Equity and diversity.
Research. I think something like that.
How do you make research diverse? Is research diverse? Does it have to be diverse?
Should be.
Why?
Come and sit down.
Come and sit down.
Come see the principal. Thank you.
So, Mike, we just started talking, and let me just grab some coffee. Hi, please come and join us. We're talking about diverse research. Should research be diverse? Should the research?
Better than that. Is your research diverse?
Is your research diverse?
Yeah, exactly.
And that scared people away, Mikey.
Do you consider diversity in your research?
Do you consider diversity? Is diversity? No. No.
Are you diverse?
Are you diverse?
I don't know. I'm just trying to find out where the bit about us is.
What does a diverse mean?
Yeah.
Are you diverse? Are you ready to talk about diversity? What's your diversity?
I mean, I'm not diverse by myself. I guess I need somebody else that's different from me in order to be diverse. By myself, I'm not really diverse because there's no one else to compare me to. I think it's only a comparative measure of diversity, isn't it?
Yes. So, do you think you stand out? Do you want to stand out? Or do you want to blend
that's
in?
the sole reason I'm wearing this shirt, I've got to be honest. So yeah.
That's right next to me, you're blending in, Mikey.
Well, exactly, and I think that shows the point. Now, here we are, day 2. What are the practical steps we can take to ensure diversity and equity in genomics programs?
In genomics programs, see, that's what we were missing.
That's not what I said. I said, what are the steps? I said...
Oh, we're missing the steps.
I said, what are the steps we can take?
Are you asking
practical
me?
Steps.
No, you tell me.
I don't know. I mean, maybe we need some people that know more about this. Maybe this person.
Listen. Hi.
Yeah. What are the practical steps we can
What
take?
can we practically do to make research diverse? Do we need to make it diverse?
I don't know.
It's just uniform. Yeah. Uniformity is good.
Yeah, I mean...
Big Brother loves uniformity.
Yes.
4 plus 4 equals 5. 2 plus 2 equals 5.
Anyway,
Challenge
we failed to get that person to come and join in. We've got to admit, we've got to chalk it up as a failure. You know, we don't have to stick on this whole diversity question. We don't. Anything.
Yes, you're right.
And only if that lady comes back that wanted to talk about diversity.
That's what
when we come to it, just
about,
don't worry. Leave it to me. I'm going to switch it. I'm going to whatever we talk about, I'm going to link it to diversity because that's my job. So you just focus on the flow of the conversation, and I will do the linking with the diversity.
Okay, that's
so let's just talk about anything. We do need some people around the table, but maybe this person. Come and join us, yes.
Come and join us, yes.
We need something to start this off.
This is a Talkaoke.
2 minutes, 2 minutes, yeah, 2 minutes. We've got Matt here in the middle. He's our trainee host. He's also a molecular cell biologist. He's a protein guy.
A protein guy.
But his name's Matt. I'm Mikey. Tell us your name, and hopefully you'll have something we can just work with.
Hi.
Hello.
Hi. What's your name? I'm Matt.
I'm Kate.
Okay. Nice to meet you.
Nice to meet you.
What do you do, Kate? What brings you to genomics?
Proteomics.
Finally! we've got someone working on proteomics. So what do you do with proteomics? Do you do the sample analysis?
We do the broadest NGS-based panel on the market with 9,500 proteins, with the readout by Illumina NGS sequencers.
So sorry, so if I understand this correctly, you're taking NGS sequencing data. Wrong.
No. We convert the protein abundance into the somamer abundance, slow of rate modified aptamers, and then read this with the NGS.
But then why do you need to convert this data in this way?
Because
what's the benefit?
You can measure 9,000 proteins in multiplex with this technology.
And you can do that with normal mass spec? With the good separation? Or good protein digest?
We see a good correlation to mass spec, but we are able to process very large cohorts and clinical trials, which is challenging to do with the existing mass spec technologies. So this is really for the large-scale, high-throughput proteomics projects.
Mike, you've got a question about proteomics.
Also, like a training point as well, like to maybe turn it into less of an interrogation and more of a conversation. Because this is a pop-up talk show. It's you know, let's just make it a bit more. Sorry, Kate. But you know, I think there's a lot of associated, you know, questions that maybe the people that are not into proteomics might want. Maybe they need to know more about proteomics. Like, for example, in the earlier session, someone came up with the idea that actually, just so that people understand proteomics a bit more, we should actually create some macroscopic sized 3D structures that you can actually fold yourself in a fun exercise at next year's festival. What do you think about that? Would that be something that would actually spread the kind of love for proteomics that I'm not really feeling here, apart from Matt, to be honest? It's been a bit every time we mention proteins, it's been a bit of a, you know, quiet session, I've got to be honest. So, you know, maybe you could ask something like that. What would spread the love for proteomics a bit more? Yeah, that's such an excellent question. Would you like to answer? What makes you love proteins? What would make other people love proteomics too?
Well, I love proteins because they are the closest to the phenotype. So they're the closest measure to understand the journey from health to disease. The majority of the drug targets are proteins, and they are really functional, and they're good measure, reliable measure of the transition, as I said, from health to disease.
So what would make all of these genomics people start loving proteins? Should they just abandon genomics?
Absolutely not. They should look at this in a complex. Combining genomics data, especially from genome-wide association studies with proteomics, has shown to improve the discovery rate of drug targets by 10-fold by means of PQTLs, protein quantitative trait loci, which are the, you understand, the variant which is the closest, which is in charge of the, which has an influence of the expression on the protein. Yeah.
So how do we make? Do you love proteins? Do you like proteins? Protein science? They don't. People don't like it. Maybe you should ask, do you like genomics? If you like genomics, sit down. Come on. So we've got genomics person, we've got proteomics person. Genomics person is coming up. Do you like proteins?
I have to leave.
That's all right.
Like proteins.
Proteins.
Yes. They're integral part of our life being, right?
So we've got 2 people loving proteins. That's 2 to 1, because Mikey hasn't said anything. If he likes proteins or not, he's been complaining. But we've got a new face at the table. And what is your name? I'll let you finish. And hi, what's your name?
Carmel.
Carmel, nice to meet you. I am Matt. This is Talkaoke, a pop-up talk show where we just started talking about. Mikey just started complaining about me because he said like, oh, he just talks about proteins and it gets very boring. And we started talking, how do you make people love proteins? And we're talking about how proteins are really related to diseases. You know, they're a step closer than all of that genomic stuff. So maybe any thoughts? Do we love proteins more than genes?
I think I love both. I love both. Genes and also proteins.
So both?
Both.
Mikey.
I've got to say, we've been talking about his P53 is his favourite. What you've said about proteins makes it much more compelling than what Matt says about proteins, that it is actually related to disease, it's closer to disease. You've never mentioned any of these things when you talk about your proteins. For you, it's just about the sheer beauty of the structure or the kind of activity of the protein structure. You've never said anything about this is related to disease or anything like that. So this is new information for me. Thank you, Kate.
I'll need to stop myself from responding, but hello, we've got a new person coming, and final thoughts.
Final thought. My God. The more of the proteomics do different comparisons of different technologies, the more we understand how they compare, what are the benefits, what are the pros and cons. The deeper is our understanding will be where they applied the most and how we can work together to kind of harmonize omics data sets for the benefit of the healthcare.
Thank you. So we need to work together, genes and proteins, all together. We've got the new. Oh, that's all right. That's all right, Mikey. Hi, I'm Matt. What's your name?
Hi, Matt. My name's Amy.
Amy, nice to meet you, Amy. This is Talkaoke. We just started talking about proteins. We started off. Mikey just will keep repeating that I'm boring, which is fine. It's not fine.
I'm not saying that you're boring. I'm just saying that not many people have got that interest in proteins, and finally we found a protein person that shares an interest.
That's all right. That's all right.
I'm more concerned about what people are interested in than, I don't think it's boring at all.
Thank you for the correction, Mikey. So we've got Margot, we've got Mikey, we've got Sanjeev, and we've got Carmel.
Yes.
And we are meant to be talking also about what steps we need to take to have equity and diversity in research. And I think just the final thought here about joining data sets and making collaboration real was a very strong point that we can start off about. How do we collaborate? How do we work together? Is there a good way or a bad way to work together? As scientists, as humans, have you ever worked together with someone else?
Yes, of course, of course.
Was it a good experience or a bad experience?
I would say overall good experience, because you both, if it's 2 people, 3 people, people are bringing different opinions to the table. And it's always good to kind of have, whether it's a difference in opinion or whether you're kind of similar thoughts, and then just to kind of reinforce those thoughts as well. So I think it's good to have.
So it's good when it's different. So can you give an example when someone was different to you and that was actually good? Because different is diverse, right?
Yeah, I mean, we've had our sales meeting quite recently, and just in like a team activity, sometimes you're building something.
Mm-hmm.
I have a certain opinion of how to go about a particular task. Someone else has a different way, and you take on board what the other person has said, and then you come to a conclusion that is going to move your work forward.
So it's about a compromise.
Yeah, you have
It's about
to, you have to
compromising.
reach a compromise, yeah.
Margot, do you compromise? Is it worth it, or does it have to be your way?
Yeah, no, and it doesn't have to be my vision for it to be a worthwhile collaboration, but I was just wondering about this. For instance, I'm an artist, so I'm not a scientist, but I recently did a project with an artist who's not, neurodiverse, and I thought, yeah, it's fine, like, you know, diversity comes in all shapes, it's fine. But I found it very challenging to work with somebody for whom maybe some things which are obvious to me, like breathing, for example, are less obvious. They're less capable of doing something, maybe like a simple task, and that was frustrating me. I tried not to get frustrated by it and like look towards, yeah, but they bring to the table a lot of different perspectives, and their neurodiversity is not what defines them. But because the project had a finite deadline and funding attached to it, I felt like under quite a lot of pressure to make it work, and I found this collaborative approach just really stressful. And I wonder whether you feel that in your areas of expertise, where you want to collaborate, you want to be open-minded, but it's hard because you've got your boss breathing down your neck.
Like that.
So, as a boss,
Have you breathed someone's neck?
sometimes.
Does it work?
Sometimes. But I lead a team of 6, and they are all exceptionally diverse. Across the 6 people, they have very different personalities, very different strengths and weaknesses, and it's about having individual approach to each one rather than a one-size-fits-all approach. Like, I know what motivates one of them doesn't motivate one of the other ones, so it's working out what makes them tick, and then bringing them together to work as a group.
So it's about carrot and stick and figuring out who likes a carrot and stick. Mikey, you like carrots? You like sticks? Like when you collaborate?
I'm more of a carrot person, I've got to say. But I mean, I think, you know, ideally and theoretically, the more diverse your team, the better the output you're going to get is, because you can get all these different perspectives. It's going to be very rich. But there is a practical issue, and I think Margot was, you know, commenting on it, that the demands of society are not neurodiverse. So, you know, like if there is a time limit, that is more. Some people respond better to deadlines than others. So, and if there is like certain outputs, that's, you know, so even if we can create this, foster this diverse approach in the workspace, sometimes the output is not, you know, demands certain kind of abilities and skills and neuro. I don't want to say neurotypical, but a certain neuro set, let's say. I don't know what the word is for it. That requires the output, the final output.
So it's about finding that sweet spot, essentially. But we've got a new face at the table. I'm Matt. This is Talkaoke, a pop-up talk show where we talk about anything and everything, but we're talking about how to collaborate. But first, what is your name?
I'm Julio.
Julio, nice to meet you, Julio.
Thank you.
So have you collaborated with anyone?
I mean, all the time, that's what I do, collaborate.
Is it easy?
I don't
how
work
do you
by
make
myself.
it work?
Is it easy? Yeah, I find it easy. I enjoy collaborating.
So it's in human nature to collaborate. It's in human nature to be diverse. Would you say that?
I wouldn't say it's in human nature to be diverse, but I think that, you know, we're sort of into it now. I mean, we have access to so much information, to so many people, that we're sort of like, yes, we have to go and look for that. I don't know if it's in our nature to do that.
So what would you do, Margot? You've had your hand raised.
I'm just curious, from what you said, Amy, like how did you find the best way of managing a diverse team? Was it a course that you did? Was it years of experience? Was it individual conversations? Whether you changed as a person? I'm just curious as to how you achieved that kind of level of leadership, so accommodating.
So I've taken like various leadership roles over my life, not just at work, but also outside of work. You know, I play a lot of team sport. So, you know, leading a team in team sport, you realize that, you know, it's really corny, but, you know, it's kind of like being better than the sum of all your parts.
And that for me is like the true definition of team, is like you are so much stronger together than you are as individual contributors, and trying to make sure that I instill that belief in my team that it's okay to fail, fail quickly, learn from it, carry on. So that's number one. And asking for help. For me, this is the absolute biggest thing, is being able to say, I don't know how to do something, can you help me? You're stronger at me than this, can you help me? Because I will, like, some of my team members are better than me at doing things, and I'm happy to ask them for help. So, you know, being open with your strengths and weaknesses. In terms of what you said about the training piece, just listening, learning, time, and also I've worked with different team members, and I've made mistakes in the way I've managed them, and I've learned from that. Right? So it's very varied, I think.
So it's about letting your team be okay with failure. Carmel, we haven't said anything yet, but do you work with people? Is it okay to fail quickly and to move on? Do you rely on others? Do others rely on you?
Yeah, so I work for the NHS, for the Genomic Medicine Service. So we actually look to integrate genomics testing into patient care as part of and we help to create pathways. So a lot of the pathways are actually created by clinicians, nurses, academics, and what I do is I just help them strategize that and kind of help integrate it into the NHS. Yeah, so a lot of,
So it's about integrating.
Integrating, yeah.
Because strategy. Big words.
Yeah,
big words.
because genomics testing is new. It's not part of everyday healthcare. So what we're trying to do is we're trying to make it as normalized as taking someone's blood pressure. So that requires a lot of us working with clinicians to educate them, working with nurses and midwives to get them up to skill with the genetics knowledge, and also trying to, again, diversify the genetics testing that is ongoing, because so I get very distracted easily, sorry.
Oh, sorry, I'm just inviting someone.
Yeah, so I work with a lot of different people because I work on their projects. It's not my projects, it's their projects, and I just help to make it real for them.
Thank you.
You're welcome.
So we're talking about how to collaborate effectively, how to bring equity and diversity into research. My name is Matt. This is a Talkaoke show. What is your name?
My name is Bhavna.
Bhavna?
I work at our Future Health. I'm head of delivery there.
That's amazing.
Yeah. Do you know about Future Health?
I personally do, but maybe explain to...
Yeah, I mean, it's basically linked to what you just said. You're talking about collaboration and the equity of, how to make research available. I can hold this for you. Yeah.
Thank you.
Thank you. And we're matching, kind of.
We're matching, yes.
I'm very much on
this
brand.
This is your place now.
I'm very much on brand with the genomics pink. I had no idea, right? Yeah, so at our Future Health, sorry, did you? No.
Okay, carry on.
So we are all easily distracted. So our Future Health is a research organization. I joined a couple of years ago. Didn't know. This is a new field for me because I'm basically from IT. I lead teams that develop software. But coming here is great because you see loads of people wanting that, kind of equal access to health data and the impact it makes. And at our Future Health, one of the things we're doing and one of the targets we've got is to gather as much diverse data as possible. So we are hitting, I think we're one of the biggest research organizations in the UK in terms of volumes of data, as well as the diversity of data for ethnicity, lifestyle, age, underprivileged backgrounds and all the rest of it. And yeah, I think there's a lot of conversation around collaboration in the panel talk earlier as well, with I think reps from Genomics, Future Health, NHS, loads of big organizations all sitting together and kind of saying we must remove these barriers. it's really interesting. You want to say something?
So what are the barriers? Can you feel any barriers collaborating with people?
So I love Our Future Health. Really, really interested to see what it comes to fruition. I think one of the biggest barriers is actually education. And one of the most important things for me is people knowing why that they're doing something. What is the purpose? And perhaps they don't understand some of the underlying science to something. It's actually like trying to be able to explain things on a basics terms. How I would talk to my grandparents about genomics and proteomics and all of the weird and wonderful things that we can do with single cell sequencing is very different to how I would talk to my PhD qualified husband about all of those things. So it's knowing how to pitch, and it's not lecturing people, it's actually making them understand. I think that for me is a really big barrier.
That's true. I just want to add on to this. It's interesting because you say that you're trying to diversify the data, and I think like even different like cultural backgrounds, like people with different cultural backgrounds have got different things that would motivate them. Like for instance, I'm from Poland, and when my mum was asked to take part in this countrywide testing, just to kind of check the general levels of health of the population, she was selected. Like I really had to, like she was gonna not do it, and I had to like really talk to her and just like explain. And I know Mike, you did the COVID vaccination test research, but you were incentivized by vouchers for TK Maxx. So I mean, I don't know, for Polish people maybe something else. But yeah, I think that having that kind of personal context, understanding of personal context, because I think in Poland we're naturally very suspicious, like what are you trying to sell me? But when it comes to data, yeah, there could be an element of that, but essentially you are contributing something bigger, so it's good to understand that.
So Mike, you want to answer that?
I'm not gonna rise to that, because I did that and for society, for community, not for the vouchers that I got.
Sure.
But anyway, I think I think you're right. I mean, just traveling around doing Talkaoke in different lands, you see that people have got different kind of expectations. And you're right. When you go to Eastern Europe, people are very much more suspicious because they've been brought up in a society where you have to always second guess and you can't trust people. But I do think because we've got the NHS in this country, there is a lot of altruism kind of built into society that you should do things for the benefit of society, especially for when it comes to health. And I think you can actually work on that sense of altruism that people have just got from having been brought up in this country.
So it's about making people altruistic and make people feel together.
Interestingly, I think I have been one of those, like traditionally, like really, I don't want to share my data and blah blah. And I got inspired from he's 14 now, he was 10. And NHS, I think he went to the hospital for something routine, and they invited him to take part in a survey for 1 year, wherein he would fill out a questionnaire, 10 questions. Every time he'd fill out a questionnaire, he'd get a £10 voucher.
Ooh.
I mean, he was the one, I won £10. But actually, he was filling out the question, and we were sitting with him, and he's feeling like, are you happy? Are you tired? How long do you play? How much do you study? How many friends do you have? So it was like a lifestyle questionnaire thing, and he was so open at kind of giving his information back. And I watched him and I was like, this is amazing. So he's got such a positive, he's so little, he doesn't really understand. And I was like, Rochik, you shouldn't be telling all this. He's like, why? I'm going to help other kids. And I was like, that's really nice. And that change, it subconsciously made me consider, am I just holding back? And I think I'm really grateful I've joined Our Future Health. And in terms of reaching out to, so for instance, what Our Future Health has been doing is it has been putting up, like going into the Halal festival and, like those different festivals wherein they can engage with people from different ethnic minorities, or going into universities where they can well, say recruit, like get volunteers between 18 and 25. Because, in health research, as they say, stereotypical data is available from middle-aged white men. So to diversify that data, I think there's that conscious, not just choices, but conscious, you could say, effort that you have to make to engage with that cohort other than educating them, right?
Yeah, thank you. And you want to run off, but give us some final thoughts?
I'm not really too sure of what to say, but...
Just what you're taking from this session, what would you like to share or maybe change about how we treat data or collaboration?
Not really. You put me on the spot a little bit, but...
That's okay, it's a safe space. It's just one sentence.
I don't really know. My mind's gone blank. I apologise.
Oh, no, I'm sorry.
Yes, yeah.
But I hope you enjoy the experience. And
yes, no. I better get back to my booth because I've left my colleague there.
You left your colleague
I left
there,
my colleague
so you need
there
to go and collaborate.
And collaborate with him and, yeah, our company, is MP Biomedical's booth, and yeah, we're a very diverse, I'm adding a few things, we're a very diverse company with a number of people from different countries, different ethnicities, and we've regularly had meetings where we'll bring in things from our kind of local culture or country or city that represents us, and that's been a really good kind of learning curve, learning about my colleagues' backgrounds and things like that in a bit more informal and kind of a easygoing way rather than in a kind of formal presentation setting or anything like that. So yeah, well, anyway, good luck everybody with your discussions.
Thank you. So having a diverse workspace is a great thing, it's an educational thing, and we're talking about educating other people, how important this gap is to address. Mikey, do you educate people?
Yes and no. I think it's about creating that space for curiosity, because I think quite a lot of people don't like that kind of thing of like, open your ear and I'm just going to shove all these facts into your head. People like to kind of be more curious and what does this really mean for me? And it's just a different way of learning. And I think to me it's more about creating that space for people to pursue their own interests is a better way, especially when you're out of a kind of academic field and into the community. You want them to actually be driving it in the direction they want to take it.
So it's about empowering people, and you want to go, so give us our final thoughts.
Final thoughts, well, it's great to be here and talking to everyone, listening to different perspectives. I'm really enjoying the day. Again, I'm very curious. So I think when you're working with professionals at work, you have to be careful about the stupid questions you ask, but I think coming here, I can go up to each booth, sit in talks, not be judged for what I know or don't know, and just take it all in. So loving it, really. Thank you so much.
Great, so it's about, thank you. So we keep an open space. Oh, please give us a final thought.
Yeah.
Everyone gets a final thought. What time is it? Oh.
Yeah, final thoughts. Yeah, keep going, I guess.
Keep going. Keep calm and carry on. Yeah, where are you going?
Huh?
So
I'm gonna go to the antenatal reflex DNA screening for Down syndrome because I work in the midwifery space.
Oh wow!
So yeah.
So it's about screening the fetuses.
Yeah.
Great. Good luck.
Thank you.
Final thoughts?
Well, thank you. I think my final thought is that something that I always like to reflect is, and let's put kindness at the forefront of everything that we do, because I think that you learn and educate and collaborate. It comes in different ways. Just be kind to people. People, you don't know what they're going through. You don't know what happened the hour before you're talking to them. They didn't deliver something on time. You set grand objectives. You plan something fantastic behind the scenes, but just be kind,
Yeah, just be kind to people. Yes. Thank you. Hello, this is Talkaoke, a pop-up talk show. My name is Matt. What is your name?
My name is Tapoka.
Tapoka, nice to meet you. Round of applause. We've got...
Mikey.
I'm Margot.
Amy.
Amy. And I am Matt. We could swap over. So, final thoughts. We can swap over then.
I really like Julia's message about being kind, and also I say to my team all the time, like, yeah, you never know what happened to somebody today. So if they're being angry with you, just take a big breath and run through it and be polite and be kind. And I liked Mike's point as well about being curious and giving a space for curiosity, because I think that's really important.
Can I just ask, what team sport did you play?
So I did rowing when I was a child, and then I played field hockey until relatively recently, and now I run with friends most of the time.
Yeah, it's pretty awesome. Thank you. We're gonna experience a change of hosts.
No. I'm happy to be on the outside. I'm gonna take some photos.
Okay,
I'm just gonna, we're just having a little swap around of Talkaoke.
A change of personnel. We're going to keep the conversation going. Matt's got to go to a Teams meeting, but we're not going to let that stand in our way. We're going to keep this conversation going. And the whole point of Talkaoke is it's kind of up to you to decide what you want to talk about, and that's you as a collective people, but it's actually just you and Margot at the moment. But if we get it right, if we get our chemistry right, we'll have hundreds of people coming to talk. I've lost my windshield. Oh, it's over here. We'll have hundreds of people because of the compelling nature of the conversation, but no pressure. It could just be...
We'll make it compelling.
Yeah, that's what I'm saying. We may make it compelling, we may not, we don't know. Yes. So, was it Tapoka? Tapoka, I've got Margot, Mikey. What have I got? I've got something on my... that's from the bicycle, isn't it? Anyway, so... Talkaoke, we've talked about a lot so far today. We've talked about all sorts of things. How to make this festival more fun was a good one. No wonder this wasn't working properly. How to make this festival more fun. We talked about science communication. We talked about proteins. Are proteins getting enough airtime, especially proteomics? Is it something that needs a bit more? I mean, to be honest, every time we talk about proteins, everyone disappears. But anyway, I'm just doing it from Matt. We talked about loads of things, but we can do. What would you like to talk about, Tapoka?
Oh God.
What are you here to do? What are you here to find stuff out, to present, to?
Yeah, just to meet the genomics community.
Yeah.
I am an omics scientist.
What kind of omics are you?
I do all the omics.
All the omics?
All the omics.
Can you tell us what all the omics are? What are all the omics?
So my team studies a parasite called Cryptosporidium.
Yeah.
And we use genomics, transcriptomics, and proteomics.
Yeah.
To understand host and parasite biology.
Right.
And also develop drugs, vaccines in the future.
Wow. I mean, can you tell us a little bit more about this parasite? Because I've got to say, I've never heard of it. What is it?
So Cryptosporidium is waterborne. You might have heard of the outbreak that happened in Devon where some water pipes burst and a bunch of people got sick.
Yeah.
That's our parasite.
Okay. Was that an experiment by you, or was that just...
That was an unfortunate experiment by the water companies.
Right.
And the more I hear Thames Water in the news, the more I get worried.
Okay, we've got a button for that. Okay, so Cryptosporidium. Margot.
Are there any benefits of parasites? Because obviously to call something a parasite, I guess it's not a nice thing.
It's got a bit of negative connotation, doesn't it?
Exactly. So my question number one is, are there any positives from parasites? And secondly, I'm seeing a lot of social media content about bad quality of British water, which I think parasites maybe is one side of it, but maybe there's some other ones, and I just wondered about your thoughts. Is it safe to drink water from the tap? If you
Okay, so answer either or any of those questions. What do you want to talk about?
Sure. I used to work on a parasite that we actually thought we would be able to use for good.
Yeah.
So I used to work on a worm called whipworms.
Yeah.
whipworms. So they had a short stubby end and a long flappy end, so they got named whipworms. But yeah, those whipworms could get into your body and kind of convince your immune system that nothing was happening.
Yeah.
And so we thought we could use those for people who have autoimmune diseases, whose immune system might be a bit hyperactive. So yeah,
just tone it down a little bit by getting extracts of whipworm.
Exactly. So yeah, there's some labs that develop that use proteomics to look at what worms, basically what's in worm saliva that we can use to help people who have autoimmune, in particular autoimmune gut issues, but maybe even things like psoriasis.
Right. And has that worked? Has it been successful?
So yeah, so there's some trials human trials in the US at the moment, which is pretty cool.
Okay, so parasites can be useful.
Parasites can be good.
But what about Cryptosporidium? Has it got any positives?
Cryptosporidium, no real
Not
benefits.
so far,
It's one you want to avoid.
Yeah. What does it do? What does it do to a human?
Very unpleasant couple of weeks, lots of diarrhea. So for you and me, if we got it, couple of weeks of that.
Come and join us. Have you ever had a parasitic infection? You don't have to have had one to join this conversation, but we've got a parasite expert here. That didn't work, did it? But anyway.
I find that normally happens. Normally I'm like,
people don't want to know about parasites until
and I work with poo, and everyone's like,
But people don't want to know about it until they've got a parasite, and then they want to know. Okay. Come and have you got a parasite? Yeah. Okay. Well, it's going in the wrong direction if he's going to the toilet. It's either north or south. But anyway, okay, so parasites, you don't want to have a parasite, for sure. You're using every omics you can to find out more about it. What is your role in all this? Are you just the omics coordinator?
I am.
So you're kind of almost like a conductor of all the different omicists.
Exactly.
Yeah.
I'm our team's sequencing expert, our bioinformatician. So I run a lot of my own sequencing projects and anything else sequencing related that someone wants to do in the team. I'm quite frequently roped in.
So you're mostly sequencing parasites. Is that different to sequencing humans?
Very. So the genomes are obviously a lot smaller, but also the resources we have are a lot smaller.
Right.
Because a lot of people haven't put the amount of money that has gone into the Human Genome Project has never gone into a parasite genome project. So a lot of what I do is I'll do all the sequencing and then be like, how can I make this interesting? What have I actually found out from these sequences? What do people actually want to know from this data? So it's a lot more challenging.
So in a way, you're doing the jobs that maybe everyone in this whole festival is doing for the human genome, but you're doing them all yourself rather than getting 20 different teams working.
Exactly that. So it's everything that we do has got the interest of studying the parasite, but also developing resources for the field. So it's if I sequence this, I can answer my biological question, but I'm going to bank some data or create a new reference genome as an aside to what else I'm already doing.
Sounds like it's a lot of work.
It is.
Okay, that's this button. Any thoughts, questions, Margot?
I'm just trying to think. I'm just brainstorming in my own brain about how to make parasite. Exactly.
Yeah.
How to make the parasite cause more out there.
You
so
want
it
to see
attracts
things
more.
from the parasite's point of view.
Well, maybe. I mean, I just wonder how to make it more fundable, more interesting, more for people to see the relevance of it.
Yeah.
And maybe this kind of the outbreak in Devon wasn't such a bad idea, just to kind of highlight it a bit. I mean, I don't know. Could we make? I mean, I know it's mixing two different things, but maybe we could make it go viral. I know it's the parasites, but
can you make a parasite go
viral? Yeah. I don't know.
Or can you make a virus go parasitic? That's another question. Do parasites get viruses? Do they have their own viral infections?
No, that's honestly the perfect question. So Cryptosporidium has its own virus inside it, and so that's one of the things that makes it a bit complex when you look at
a human or a mouse immune response. Sometimes it's not the perfect response you're imagining, and that's maybe because of the virus inside the parasite is also affecting the
Okay, so is it just one virus? Is there many viruses? And what does it do to these poor little parasites?
So they just have one virus. We don't know that it does anything to the parasites. It's just been a passenger with them for quite a while now.
Yeah, okay.
I kind of wonder, you know, sometimes when you've got like a homeless dogs shelters or whatever, it's like, oh, take the dog in, you know, help out. Could you have a pet parasite? I'm trying to I'm still thinking how to make it more, you know, so people care. It's like, oh my, you know, at some point when I was a teenager, it was very trendy to have a stickworm. Sorry, not stickworm, stick insect. Yeah. Now that's not your classical type of pet, and yet people did it because it was like a bit quirky. So I wonder whether, you know, pretty niche, but.
I mean, do you keep parasites in the lab, and do they have names or?
We try not to get too attached to the science.
Okay.
Yeah.
Okay. Can that be an issue? Sometimes you get too attached.
Yeah. No, because. So a lot of our parasites, because we can't just grow them in a dish in cell culture, we have to grow it in an animal model.
Right, okay.
So yeah, we're not allowed to name the animals because
you're not allowed
sometimes they do get quite sick from the parasites.
Okay, but the parasite itself, you don't.
The parasite itself, no, we don't.
You don't give them a name either.
No.
Okay, and what kind of animal do you grow these parasites in?
Cows and mice.
Cows. I mean, that's quite a large. I haven't got a mouse sound effect, but I have got.
It's a very good mouse
okay,
sound.
that's the cow one, and do the mouse. Okay. But I mean,
it's gotta be wrong
yeah,
in my life.
Yeah.
Gotta be like
I mean, it sounds like quite a hard ask to grow a parasite in a cow. How do you get do you just go through the cow's excrement to get the parasites and get them into the lab?
Yeah, no, that's pretty much it. We partner with farms, so because oftentimes these cows have this parasite anyway.
Yeah.
And yeah, it's literally.
Okay, so they've got it, so you don't have to plant it in there. They've got the parasite.
So yeah, so we
come ready parasited.
Exactly, but yeah, it's just we don't have to do the work. The farmers collect the buckets of poo and ship us the parasites after they've been filtered out.
Is there a lot of excitement when a new shipment comes?
Oh, absolutely. Everyone's like, new parasite day. Everyone's got their experiments planned and is like, I need this many parasites.
Right. Okay. Is there a bit of a fight over who gets the cow patch?
Yeah, absolutely.
Okay. Right, and where is this happening? Is this happening in London or in the countryside?
Yeah, no, we're in London, so I'm based at the Francis Crick Institute.
Okay. We've been there with Talkaoke quite a few times.
Oh, awesome.
So look out for us next time we're there.
Yeah.
In fact, you could even ask the science communication people to remind them that Talkaoke does come to the Crick every now and often. Isn't it about time we came again? But yes, we've been there. We've been doing that job as well of actually being outside of the institute, talking to the passers-by, a bit like this person. Although I'm sure you've got some kind of knowledge of genomics or something. You do. There we go. That's we have got Margot, we've got Talkaoke, I'm Mikey. Tell us your name.
Vaidehi.
Yeah. And when we're talking about, because Talkaoke's a parasite expert in the field of Cryptosporidium. Have you heard of this?
I'm not good with organisms.
No?
I'm a bioinformatician, so I just work with data analysis and all those things.
Okay. All right, so maybe you could explain a little bit about the Cryptosporidium parasite to Wahida.
Yeah, absolutely. Our parasite infects intestinal cells, and so we sequence a lot of samples from mice, humans, and cattle to understand the parasite biology, the host immune response, and everything in between.
Sorry, I'm just backing you up there. Do you ever need a bioinformatician to kind of?
I am our team's bioinformatician, so yeah.
You are the bioinformatician. So you do, not only do you coordinate the cow patch, you actually also coordinate the data as well.
Yeah, absolutely.
Okay, what's easier, cow patch or data to coordinate, and what's easier to store?
depends on the day.
Yeah, okay. So some days...
some days are good lab days and bad desk days, and some days are bad desk days and good lab days.
Okay, so in a way you've got the best of both worlds. You're working with, like, let's say, wet organic material, and you're also working with the dry and zeros and ones and the data as well.
Absolutely.
So would you like to do that? Would you like to have a kind of more of a...
Yeah,
a foot in the laboratory?
I have some experience of working with cells in bachelor's, so if I get a chance to work with cells again, I might. But yeah, I prefer dry lab more.
You like it dry. You wouldn't get your hands dirty trying to find parasites in a cow poo.
It's really complicated.
Yeah. It sounds complicated to me. I mean, where are we going to go with this conversation? We've now got Margot, we've got Wahida, is that right? Hey, is that we've got Talkaoke, we've got me, Mikey, maybe this person, yes. Join us 2 minutes, maybe not. Where are we going to go? We're running out of time in the Festival of Genomics. Yes, join us if you want, but we are running out of time. We've got maybe 2 hours left. What are the crucial things that we need to talk about in this time? What would you say? What do you think we need to actually get off of our chests today? You look pretty happy, so I'm gonna. You look like you've got nothing to get off your chest at all. You're happy just doing data, doing bioinformatics. What can you explain a little bit more about what you actually do?
The main work that I'm doing is single cell and spatial.
Yeah.
So I'm doing data analysis for liver tissue and lung tissue.
Right.
Cancer cells.
Yeah.
That's what
But
I'm
what are you
working on.
doing? What you have to explain that a bit more for people like me and Margot that don't really understand.
Yeah. So basically.
Hello. Hello, hang on a sec, but just before you explain it, we have got Margot, we've got Wahida, we've got Talkaoke. Tell us your name.
Abdi.
Abdi, and I'm Mikey. And we're just about to find out, you're doing, Wahidi, here's a bioinformatician, yes. Maybe this person is as well. Are you a bioinformatician? Or are you... Come and sit down, come and get involved in this. You're just about to explain what you do, something about liver cells and lung cells. You're doing something with that, but can you explain what you actually do?
Yeah. So basically, when we're working on 10x Visium, so when the RNA sequences are sequenced, and they extract the data, the data comes to us and we analyze. So what are the cells involved that have, what are the tumor causing genes, what are the tumor, non-tumor markers and all those things. So basically analyzing the data that's sequenced.
Okay, so you're sort of finding out what genes are actually at play in some of these phenomena.
Yeah.
Okay, we're going to find out, so we've got Margot, we have got Wahida. Wadahi. We've got Talkaoke, we've got Abdi, and we've got
Martin. And
Martin.
This is Pep. This is Pep the penguin.
Pep. Okay. And I'm Mikey. I don't have any toys or any pet penguins. But anyway, so we're just finding out what people do. We're finding out. We're running out of time here at Festival of Genomics. The clock is ticking. What do we need to get out in the last couple of hours? What actually do we need to talk about? I was looking for, like, you know, some kind of sense of, like, things you need to get off your chest. But Wadahi is very, very happy analysing cells. Abdi, have you got anything that you need to get off your chest? Any thoughts that you feel like need to get out?
Yeah, it's something very important. Sadly, I don't know how to get enough swag. Like, I've been walking around.
Right, okay, what are you looking for?
I need more notebooks, more pens, some sweets.
Okay, so we've got these mints.
That's the one thing I don't want.
Okay, that's... Okay, well, you're looking for notebooks and pens. Do you want to get more stuff?
Yeah.
A pink, do you want a pink?
I'm looking for a pink tote bag. I don't know where it is. I see people with it.
I think that comes from Amazon. That comes from Amazon.
No.
Doesn't it? No. Can we get one of these penguins?
You can if you are very nice to me.
Okay, whoa. Abdi, we've just upped the ante here. What is tell us about the penguin. Why have you got a penguin?
So this is Pep the penguin. This is Cambridge Bioscience.
Yeah.
New,
what does Cambridge Bioscience do?
So we're a distributor. So we distribute for about 60 international companies across the whole of life science research. Includes lots of products, but specific to this, we sell lots of molecular biology products for nucleic acid extraction, NGS, library preps, services for NGS, and Pep sound your little.
That's
friend.
your mascot.
It is. Yes.
Yeah. Okay. All right. So how's business gone this
It's been very good. It's been fantastic. Yeah. And we also have pick and mix at our stand as well. So,
really? Okay. So you've got lots of things. What I mean, are there different kind of things which attract different types of scientists? Like, for example, a bioinformaticist, would they be interested in a different kind of offer than, say, I don't know, like a geneticist or something?
So bioinformaticians are kind of using the data that's produced by the products that we sell.
Yeah.
So the NGS people would use our products, produce all this crazy
what
data.
What kind of freebies do they like? That's my question.
Freebies? Wow, sweet. Pick and mix.
Okay, that goes for everybody.
Everybody.
And what I'm
Who
trying to
doesn't
find
like
out,
sweets?
is there a difference? Is there a difference? I mean, what are you, Abdi, what's your kind of role in all of this?
So I know nothing about life sciences. I'm just here to talk a bit about IBM. I've just been brought in because I know a lot about AI, and everyone's talking about AI.
Okay, so you're an AI expert.
Yeah, I teach, I research, and I sell AI part of IBM. So we talk to a lot of smart people about how they can use ChatGPT to answer some questions. That's what I'm here for.
Okay, right. Okay, so you're just here. I mean, it doesn't really need selling, and I've got to say,
that's true.
It's been one of the main subjects of conversation, AI. But you're here are you here to kind of make deals selling your AI, or is it just about getting people more excited about AI?
Yeah, a bit of both. So I work for IBM, a big tech company. So a lot of people have heard of them. Yeah. So I just help sell that platform to customers. Outside of that, I also teach. So there's a lot of students here I know, so I'm also telling them about things they can do to improve how they learn and understand AI. Yeah.
All right. Okay. I mean, where do we want to go with this conversation? Do we want to talk about freebies? Do we want to talk about supplies? Supply. You want to talk about supplies?
Oh no. I was going to say, I will prepare my finest bribe. What would you like for the penguin?
Okay, you want the penguin. Yeah. Does Talkaoke need to sign a contract? She works for Crick Institute, so I'm sure they're a big customer of yours. Does she need to sign a new contract?
I'm the account manager for London, so Francis Crick is one of my best accounts. So there you go. Go to booth 117 and you can get a small PEP.
Okay, so you don't get the large one, you
don't
get a
get
little
a big one, but you can have a small one.
Okay, right, okay.
But you need a voucher.
Okay, well, it's probably easier to put in a bag a smaller penguin.
Thank you.
Okay, so, yep.
Has a question to ask.
Okay, yeah.
Thank you. Yeah, but I'm behind him, so I understand it's hard to spot. But you know about the mascots, right? First of all, it made me think of the Duolingo owl, and I thought, maybe that could be, because people were saying earlier, but how do you get people who don't understand science to, how do you incentivize them? And yeah, Duolingo for science would be one of my things.
What do you mean, like a Duolingo language app for finding out what scientists are actually going on about?
No, but it's like in Duolingo, when you learn a language, it's like, you start small, like, is this a molecule? Is it a blah? I don't know, whatever. Just like, and then you build it, and it's like, I think you can build it. You can build your understanding of science using an app. That's what I'm saying. But the second thing I was thinking, we've had a lot of conversation about poo samples, and you brought the cow pads and stuff. Why is there no mascot of that, emoji of the poo, the smiling poo? That should be the emoji. That should be the mascot. I haven't seen them around. I think it'd be immensely relevant.
Maybe next time you come along to Talkaoke, you need to come with some
smiley poo.
Yeah, some sort of cuddly poos that are poo emojis, just to promote what you're doing.
It's something we thought about. So in the team I used to work in, one of my colleagues actually wrote a comic book called The Adventures of a Scientist and Super Poop. So we had a little rainbow poop mascot, and we were teaching everyone about the gut microbiome and all the friendly microbes that are in there, hence the rainbow poop. So
you've been there, got the t-shirt, basically.
But yeah, Adventures of Super Poo. I don't know if it's in the public domain, but it's out there somewhere.
I mean, do we all need to be like Martin and have a mascot of some kind? I can see you with a cuddly poop, a cow pats. Do you need something to kind of, has IBM got any animals, any characters? I mean, Microsoft's got a paperclip. You've got to have something, surely.
We have a bee. So for IBM, the bee is meant to be a little bee.
Right, I see.
But creative, right?
Yeah.
Yeah.
Okay, let's get a final thought from you. Thanks for joining us. Thanks for joining us.
It was fun meeting you all. I have a lecture to attend, so I'm just gonna leave.
If you were going to have a mascot, what would it be? I mean, would it be like a liver because you're working on liver cells? Would it be like a cuddly liver?
Well, most of my work is on RNA, so I'll have a cartoony RNA maybe.
Okay, like a kind of chain or something like that, like a little, could be a key ring or something, like an RNA chain on a key ring. And if you've got a lot of keys, then you get a DNA chain. I don't know.
Yeah.
Yeah, could be. Okay, thanks very much.
It was fun, thanks.
Thanks for joining us. Did you have something to say, Margot?
Yeah, thank you. All the conversation about poo and you talking about working in AI, is there some kind of like an equivalent of poo in the world of AI? Like, is there anything, I mean, you know.
Well, they say garbage in, garbage out. What is the garbage data, the kind of excretory data that, you know,
is there something to be gained from bad data?
Spits out that it, you know, in a way it just wants to get rid of?
I don't know, maybe anything on Reddit? That might be poo data, I don't know.
Is Reddit that bad? I thought Reddit was quite...
I use it all the time, so...
Yeah.
Just joking, I use it all the time. Yeah, I think all data can be clean, though, so I think you can find ways. You know, the scientists in the room probably know that you can do something to it to clean it up.
Yeah.
You know,
is there a way
to
talking about, you know, data as feces, is there a way to compact the data? Because...
Compact?
I mean, to be honest, this year we've not had it, but normally we get all these bioinformatics people complaining about the data just kind of getting bigger and bigger and bigger.
It's good. I work in tech. For me, that's good.
Okay, so you want more and more data.
Yeah, it just means you probably have the wrong ways of storing it and accessing that data.
So you just want to sell more storage?
Yeah, it's pretty cheap now.
Okay.
You can't find...
That may be why that's why people stop complaining about it. I don't know. But yeah, 2 years ago, everyone's complaining about all this data storage. Now they don't care anymore.
Yeah, no, now it's gone a lot cheaper. Like if you go to Amazon or Google, you'll find S3 buckets where it's very cheap to just plug your data in. So I think data stores has gone cheaper, especially because of AI and the vast amount of data you need for that. Yeah, I don't know if there is poo associated to AI. I don't know.
Okay.
Unless we're analysing it.
Okay, well maybe that's something to think about and come back next year. You'd be like, oh, I came up with the idea of, you know, what is, when it comes to AI, what is the equivalent of computer poop? Anyway, where are we going to go with this? Is it something to talk about, computer poop? I don't know. You're happy because you've done a lot of business, you've sold a lot of whatever you sell, I'm not really sure, something to do with suppliers. I'm just going to say suppliers. Yeah, suppliers. It's been a good couple of days for you.
That's been fantastic.
Yeah. Have you come up with any ideas for new products from people?
Well, we don't make our own product. We sell other people's products.
Okay, so any new products to supply that maybe you don't supply?
We're always looking at distribution.
Okay, so it's all worked out fine.
Absolutely.
Okay.
Good. Thank you so much. I'm going to have to take Pep to the chill out zone.
Okay,
well,
we might come back. What is it, 117?
Booth 117. Come get some pick mix.
Right, come and get our penguin. Okay, now you're driving it all over our traffic away. But I want to thank you, Martin. I want a final thought from you and you. Give us a final thought. Talkaoke, give us a final thought.
Yeah, I think Talkaoke, I didn't know what it was, but I'm glad I stopped. It's been a lot of fun.
Yeah. Don't forget to recommend it to the crick, and that also includes Cambridge Biosciences and IBM. Don't forget to mention all those things to all those the purchasers of the crick. Let's get a final thought from you, Abdi.
Final thought. Big data is great, and yeah, I hope everyone has a great weekend. I'm tired.
Is it just going? I mean, you're saying more and more storage and more AI and more meta
just more
analysis of the. Is it just gonna? Are we just gonna exponentially just have more and more data? Is that the way you see it?
Hopefully, I mean that's how these models get better. That's how we can automate more things and have better AI systems in the future and make our lives easier. So we're not doing things in Excel or writing emails. Let's just automate the boring things away.
Okay, so all the tedium will be sucked up by the AI.
Yes.
Okay, you heard it here first. Thank you. Maybe it's time just to take a little break.
Just a tiny question, just because some hours ago we were talking about sense of humor. When you represent a big institution, you can't really use sense of humor to kind of promote your product or your services or whatever, because it makes you look unprofessional. And you've got I just wanted to ask big organizations around the table, what's your stand on humor in your line of work?
Okay, can you use humor to talk about what you do? You're all representing huge organizations in the field. Can you use humor, Martin?
Absolutely. I've been a sales rep for 25 years, and some people take it far too seriously.
Okay.
You have to go at it with a sense of fun. We're selling to human beings. We're people selling to people. So I think a sense of fun, absolutely.
For now, until Abdi gets his way, and it'll just be AIs selling to other AIs. Yes. AI-generated gags. I think when AI cracks comedy, then we're really going to be in trouble. So this is Talkaoke Pop-Up Talk Show. Come and sit. I've just cleaned this bit, so it's nice and clean. Well, relatively more clean, let's say. This is Talkaoke Pop-Up Talk Show, where we just get people talking about whatever they want to talk about. It's kind of up to you. We've talked about a lot of stuff. Can I have that back? Thank you.
Yeah, I mean, we've been talking about a lot of stuff. There was a lot of, obviously, scientists on the table, and there are probably still. And that continues. That seems to be the thing. I mean, may I ask you what kind of what kind of scientists are you, and what kind of stuff do you do? I'm just curious.
Yeah, sure. I have worked in cardiovascular, so heart. I studied a lot of heart cells, what happens in heart failure, and how we can treat it, and worked mostly on tools where you can use those tools to target genes which are certain genes which are certain part of your DNA, which you can chop pretty precisely.
Right.
And treat heart failure or heart diseases.
Are you involved in this as well, Harish? You seem to know about it.
No, I'm not.
Oh, okay. So you are involved in it?
Yes. I have to
are you a scientist as well?
Yes.
Where did you meet? Did you meet in a science place?
Are you both scientists or non-scientists?
I can say myself.
Because I can then
I'm not a scientist, no.
Because then I can choose the words.
Yeah, choose the words. Choose an unscientific words.
Live words.
Yeah, yeah.
So yeah, I'm Nareshan Chate. I'm a neuroscientist.
Okay.
I work at University College London.
Okay.
I have my own research group.
Right.
At the Institute of Child Health.
Right.
So we're trying to understand the disease that affect the children. And the brain disorders, both the disorders that affect the mind, but also the circuits, the molecules and cells that help us.
Is there a genetic component to your research?
Quite a lot, actually.
Yeah.
Because there's genes which are important for brain development, and if the proteins they're making, if they function 100%, we have a normal development of brain.
Yeah.
But if the proteins are not functioning at the full potential, certain proteins, then the development, the connections is compromised, and that can lead to certain disorders like autism, for instance.
Right. I mean, is it one particular gene that you've identified, or is it like this whole cocktail of different genes that I've got?
So my work, the group that I work, my group does is looking at circuits.
Right.
So we don't necessarily focus on a single gene. But the genes that are working together in a circuit that are regulating a certain function, like let's say stress, reproduction, appetite, circadian rhythms, social bonding, so this kind of stuff actually, which is important.
Yeah.
So there is both focused very targeted on certain genes, but also we'll look at the whole transcriptomes to get a broader picture of what's happening.
Okay, we've got a new face. We have got Margot, we have got Katie, we've got Harish, we've got
Simon.
Simon and Mikey. At the moment, we're just finding out about Katie and Harish's research. Whereas Katie is a heart specialist, Naresh is a brain specialist, but they are a couple. So I don't know if the brain. Yeah.
And there's no hard feelings.
But anyway, we're just finding out about that. We're finding out about it's about neurodevelopment of children, that sometimes they don't have the right proteins for their brains to develop. Naresh is researching into that, the Institute of Child Health. Simon, what brings you here? Either to the Talkaoke table or to the Festival of Genomics.
I don't know why, yeah. So the festival, I'm in industry, work for a small biotech company.
Yeah.
And one of the things we're looking at is developing genomic signatures of response to our drugs.
Right.
And I run the team, but I don't know an awful lot about genomics. So one of the things I'm here is to find out more about it.
Okay. Well, I'm not going to tell you anything about genomics, but can anybody around the table tell Simon about genomics and maybe how can he use this science to find? Was it about the efficacy of your drugs?
Yeah. Who should go on to our drugs and who perhaps should go on to an alternative?
Okay. Have you got some?
So there are a lot of genes in the genome.
Yeah. Yes.
Which are implicated in processing drugs.
Yeah.
So these are called. There are many pathways like cytochrome P50s. So what they do is basically they break down the drugs.
Right.
And certain individuals may have certain modifications in the genes that makes the gene function maybe too fast, maybe too slow.
Right.
So if a person is having a gene that is functioning too fast, then that drug will be metabolized too quickly.
Yeah.
So they might need a higher dose to have an effective
We heard about
response.
this with coffee earlier on, that some people can metabolize coffee. I know caffeine is probably not one of the drugs that you sell, but it is one of the. These guys over here need to listen to this. Some people metabolize caffeine very quickly, other people not so quickly, so they get more of a hit from the coffee.
I have to have my coffee at least before noon. If I drink afternoon, I can't sleep.
Okay, so you're a slow metabolizer. Yes, come and join us. So we've got Simon, we've got Naresh, we've got Katie, we've got
Sally.
Sally.
Cancer biologist.
Okay, and we've got Margot, and tell us your name.
I'm Sandy.
Sandy, and I'm Mikey. At the moment, we're just finding out, basically, Simon has come here, a bit like us really, not knowing that much about genomics, but you know a lot about drugs, and you've got drugs that you produce, but you want to know, can genomics actually help me to understand the efficacy of my drugs? We just heard from Naresh that actually you can find out from genes from genomic studies, how well people process drugs, like, for example, caffeine, but there are other drugs available. And that's where we are right now. So, well, that's exactly what I'm doing. Sally, have you got any views on this? What brings you to the Festival of Genomics?
I'm just here around. Actually, a bit curious about the AI development and how it combines. And then I know there's gonna be a talk about drug discovery and AI over there later, so probably
Okay.
And so we're talking about.
I mean, right now we're talking about, can you use genomics to actually understand how effective your drugs are? But maybe I was also ask Simon, as you're a drug guy, do you ever use AI for drug discovery? Are you thinking about using it?
Certainly thinking about it, and it's a question of a lot of hype, and I think we're gradually getting through some of the hype, and seeing what's really useful. Absolutely, when thinking about how we can use it, we're a small molecule company, and there's a lot done about design of new drugs, and I think that's an area. There was a good talk on it just before lunch I went to, and I think that's an area that's really settling down now that we should incorporate with us, and we're thinking about that now. We're a small company. On the biology side.
Okay, so you don't just do small molecules. You're a small company that.
We're a small
You're not
company.
a small molecule company.
No, we're a small
You've
company.
You've got like a maximum number of atoms in your molecules.
Small companies, small molecules. Yeah.
Right. Okay.
Yeah. And I think that's an area that's really good. The biology side's much more difficult. I think.
Yeah.
You know, a lot of people looking at it. How can they use AI for all sorts of different things to do genomic analysis? That's a bit more difficult to work out how to work it. How to do it.
So start off with the smaller molecules and grow upwards, Naresh.
So do you ask ChatGPT to give me a good drug that works for X, for instance, or how do you apply AI in drug development?
Is it simply asking ChatGPT, or are there other AIs?
Are you asking, like, you put a chemical and say, can you give me a better molecule which is long-lasting, for instance?
The most examples I've seen have been sort of trained on some data set. You know, there's an example for lunch, a fairly classic, deep neural network or something where you have a training set. That's, like ChatGPT, you put on it, and then you can say, okay, design me a molecule that fits into this protein space, something like that. And the interesting thing was, it said, instead of taking just a library of known chemicals and saying, okay, we know we've got 2 million chemicals in our library, which one will stick? It'll design your own new drug as well.
Yeah, so it'll create a completely novel molecule.
Completely novel drug, and then you can filter it and say, that's going to be toxic, that's not going to work, but these bunch might work.
But I mean, if you've got this kind of novel molecule, is it, from your side as a sort of manufacturer of molecules, can you actually make these things that the computer's coming up with?
Yeah, amazingly you can. Yeah, you have to work out whether they're stable and so on. Make sure they're not, not got known toxicities. But other than that, the chemists are amazing. They just cook these things up. Absolutely amazing.
Okay, so we've got a new face at the table. We've got Sandy, we've got Simon, we've got Naresh, we've got Katie, we've got Sally, we've got Margot. Tell us your name.
I'm Matt. Hi.
Matt, we're talking about AI and drug discovery, AI coming up with novel molecules that fit into it's a bit like, going to the shop and saying, have you got something that fits this? You just basically find a receptor and you just ask the computer to come up with a new molecule. Come and join us. Come up with a new molecule that fits the space. Apparently AI can do that. It's better with a smaller molecule, I think. And Simon has got a small molecule company. So that's where we are right now. We've got a new face. Tell us your name.
I'm Victoria.
Victoria, welcome. We've got Simon, we've got Naresh, we've got Katie, we've got Sally, we've got Margot, we've got Matt and Sandy, and I'm Mikey. So that's where we are right now. But you're saying as well there's a little bit of hype going on there, but that hype is actually, it's almost like fake it till you make it. It's actually starting to turn into real results.
Yeah, definitely. I mean, any new technology, people have got to get funding. I don't mind at all, and Matt wants to have a go.
Yeah.
But I think at the end of the day, when all the hype's over, people say, okay, show me this.
What we actually left with.
Yeah.
Now we've got the machine that we got the funding for.
Yeah.
What can we do with it? Okay. Yeah.
As a scientist who works in proteins, one of the greatest challenges, I think, is because I think that the model of having rigid molecules that fits a rigid body of a protein is only applied to well-folded stuff.
Yeah.
Most of the proteins that we have in our bodies are intrinsically disordered.
Yeah, it's a bit like my bedroom. The clothes could be well folded, but generally they're disordered.
Generally they're disordered. So the problem is how do you drug something that doesn't really have a structure?
It's more squidgy, it's more floppy, and
how can we make molecules? And I think that's one of the great gaps that I think the AI cannot fit very well yet, because it just doesn't have the data.
Right.
So we as scientists, we need to come up with new ways.
I mean, is there a way to actually get the squidginess into the AI model?
I think some people are working on it, but it's although people have been talking about this for 20 years or more than I am alive.
Yeah.
Definitely, but I think there's not as massive models as AlphaFold, for example, which only deals with well-folded stuff. And yes, you can do these searches, but my point is, we need.
You could have just said squidginess.
I could have
saved yourself a lot of breath.
But
yeah.
What I mean to say is that we need new data.
New data on squidginess. Victoria, have you got any? Because I feel like I've been enjoying you and Sandy. Have you got any views on this? Do you feel like AI has got the promise? What brings you to genomics? Are you?
I think AI is very clever.
Yeah.
Yeah,
that's definitely
I'm afraid it's very clever and very unreliable.
Okay. Well, a lot of clever people are quite unreliable, so maybe it's just following that model. Yeah.
So I think for AI, the key is to have enough structured data to train anything, but I always find that, especially in the scientific field, the data is not structured and it's not harmonized. So, some people will call a sample, some people might call it a source, and all of the data points have different names, and it's so difficult to train to do machine learning if you don't have structured data. So I think we need to go to the root, and before we try and get very creative with AI and using AI in science, I think the fundamental is having a good data model and harmonized data sources and harmonized data fields.
Can't AI itself kind of tidy it, a bit like my bedroom, tidy it up?
No, unfortunately, that's not what AI can do. If you tell it to do something, like you teach it how to do it once, and then you tell what to do, it will do it. But without you teaching and without you empowering it with the right knowledge, it cannot do anything. So. And that right knowledge comes from the data source and harmonization of data.
Okay, so the data's not harmonized enough. We've got a new face over here. Let's quickly get your name.
I'm Molly. I was here earlier.
I know. But this is part of the protocol here. We've got Sally, we have got Katie, we've got Naresh, we've got Simon, we have got Victoria, we have got Sandy and Margot and Matt, and I'm Mikey. Who was I going to give the mic to? But you, Victoria, was it? Yes.
I was just going to say that you can't put the data on there for ethical reasons. I think that's the central issue that is faced in medicine. Otherwise, everyone's rampant with our data.
I mean, is there once again... Okay, Sandy's saying... this is a question to you, Victoria. Sandy is saying you can't use AI to tidy up your data. It needs the tidy data to begin with, standardized, harmonized. But can you use AI to help you with things like consent and ethics? Can it actually create a dialogue with those people that have given that data?
Yeah, it's really good with that. But I think that you can't put any data on there that will identify people into those big...
Yeah. I mean, what do you think about that? Do you think that's right or that's wrong, or
I think
it's just how it is?
I think that that's right, and that's just how it is. But I don't know whether you're going to be able to get safe, like proper sort of AI, powerful machines that can take medical data in a safe way.
Okay, it's a challenge for AI. We had a guy called Abdi here earlier on who's working for IBM who would have been perfect for this. But can AI solve these challenges? Can it actually tidy up its own data, ethicalize or create a consent model for its own data? Naresh, what were you going to say?
No, I was just touching upon, we're talking about ethical and sustainability, right? So now everybody's using AI. So, for instance, before we used to use Google for normal search, but now using AI, which consumes a lot of power.
Yeah.
So basically, to go to a supermarket, we're using a Lamborghini, for instance.
Right. Okay.
So what about the power on all the, where is this happening? The electricity and this, it consumes a lot, right?
Yeah.
So what about if everybody starts using AI? How are you going to support this amount of processing
Okay, so
power?
you're concerned about the actual
climate and
energy
there's so many
resources.
questions that are associated.
Okay. Are people concerned about the energy resources? Yep, Matt.
I saw recently that people are trying to combine AI with neuronal cells, and the biggest selling point was actually saving electricity, because obviously neurons, your brain runs
just
on
needs glucose.
Needs glucose, so you know, put a cup of sugar and it runs.
Yeah.
So maybe we just need to do something. I don't know if it's ethical or not, but grow just slightly bigger.
People have talked about contributing their data. Would you contribute a few of your brain cells, your high-quality neurons, for an AI? Would you actually give some tissue?
Pardon?
Have you got spare?
Oh, do I have spare? Oh, that's a good question. For 3 years of smoking and drinking, I don't know if I do.
Okay, all right. Maybe we can ask that to other people. Molly, have you got spare neurons that you would contribute?
At the moment?
Yeah.
That was a joke. I don't know. I don't know about AI. I feel like The energy thing is a major one, but like we want it to do things that humans don't enjoy.
Yeah.
Or will save us time. We don't want it to do things that that humans either enjoy or we generate more value than an AI would. So like we want it to do like laundry or dishes or like data cleanup. But innovation and like creation, I just don't think it's.
So what you're saying is that's something that we really enjoy doing. I mean, you were here earlier on.
It's part of the human experience. I don't think that we should be outsourcing that to machines, especially the energy cost.
Okay.
Yeah.
So when it comes to innovation, creativity, do we really want to? It's okay with the tedious stuff like tidying up the data, or configuring 2 million different molecules. But what about the fun stuff? Are we in danger of delegating the fun stuff to AI as well?
On the sort of creativity, the few times I tried ChatGPT and said, do my homework for me, can you write a business proposal for X? You get a pretty, it sort of gives you some ideas, but they're all ones that you knew anyway. So yeah,
it's quite generic.
If I sat then thought about it, because all it's done is scraped 100,000 business proposals. So I think I think where it is on the creativity thing, it can get you away from a blank page. So if you're standing at a blank page with a pencil, not knowing what to draw, it can give you some ideas. If you want something truly creative, it'll probably, you'd probably have to do that yourself.
It's a bit like the assistants in a art workshop of the Renaissance, that it will sketch out the shape, but then the master has to come in and do the actual face. So the feeling is, just to answer your question, Molly, is that AI is not that good at creativity. The really fine, that innovative stuff is still in the domain of humans. What do other people think about that? Sally?
So like, for example, in cancer or like other disease, what AI is really like people are focusing on right now is using AI to help to diagnose.
Yeah.
So it actually helps to save some of the results, but the point of using AI for diagnose is, do we have enough data to teach AI to make it like more reliable as actual doctor could be?
Yeah.
And there's actually one quite funny experiment done in China. Is they use ChatGPT and compare with real doctor, and then the patient come to ask like, oh, this is my symptom, could you give me advice? And then they'll rank the doctor and ChatGPT, and the ChatGPT actually get the same mark as the doctor.
Right. Okay. So it didn't get better, but it didn't get worse either.
Yeah. So it's kind of like. If we have enough data to support AI for deep learning, would they one day actually could have the first defense of like being diagnosed or helping with the doctors?
Yeah.
Yeah.
And what about other large language models? I mean, we heard earlier on about a new one that's been developed in China.
Yeah.
Is that even better than ChatGPT? Or you don't know about that?
I'm actually not sure because it's coming out just now, and I think they use 2 different type system.
Okay.
Developed. Yeah.
Okay. All right. Okay. So diagnosis is a definite yes for AI. Are you, Sandy? You wanted to say something or
final thoughts
about?
Are you off? If
I was going to
anyone
leave,
that's going to leave, just give us a final thought because it's normally a good one. It's normally a good one.
I think AI needs to start kind of doing a lot of repetitive tasks that we do, so we can go and innovate new stuff and then train AI again to, once it becomes repetitive, then it could, you can hand it over to AI. It's like your assistant.